All right. Good morning, everybody. My name is Michele of Sloan. I'm with the Foundation to Fight HABC, co-founder, and we are here at the ULF conference in Chicago, and we have a mini session on HABC TUBB4A gene mutation, and we have a fairly condensed schedule this morning, and we had to switch things around a little bit. I will do some foundation updates shortly after Dr. Vanderver does her presentation on status of her research. So with that, I'd like to invite Dr. Vanderver to come up. Thank you.
And I'm sorry not to be staying for the whole session. It's my 27th wedding anniversary, and I try to be home for the important things, and so I'm going to hop on a flight and fulfill my promise to my family and actually come home. So my presentation is really more meant to be a structure and a framework. I'm happy to be interrupted by questions. It's more meant to just give us some structure for discussing, but please don't be worried about breaking things up. Now that I'm seeing who's in the room, I'm recognizing that I don't need to do as much introduction of HABC than I thought I did. Sometimes we get new families who are new to the diagnosis. Michele, is there a way to know if the families who are online are new to the diagnosis and it's still worth my going through sort of more introductory slides?
I think a little bit about, yeah.
Okay, because you're recording and it might be viewed. Okay, so for those of you who are very, very, very familiar and who've heard me speak about this before, I apologize for the reruns. All right, so before I get started, I really want to thank the many members of my team and the members internationally who we work with on HABC. The work that you're going to see is not at all mine alone, and I don't want to take full credit for it because we get a lot of help. So importantly, Mario Vanderknapp first identified HABC, and in the beginning we really thought it was a very sort of simple disease, right? Patients would have hypomyelination, they would have atrophy of the basal ganglia and cerebellum, their MRIs all looked very much the same, and we thought we sort of understood and knew what it looked like even before we actually knew what the genetic cause was. Now Mario was very, very good at doing pattern recognition because it turns out that most of these patients actually had a single gene mutation, not just a single gene, but actually a single mutation in the gene. And so she was very good at recognizing those features. But over time, we also realized that the typical picture, which included toddlers who would typically have normal developmental milestones for some time and then lose skills, right? And we thought was clinically a homogeneous disease. Is there any way to limit the feedback, like the feedback loop, so I'm not hearing myself? Okay. And again, you know, this specific clinical picture, all the patients had one single mutation, but over time, we realized that it was a much broader clinical phenotype. First of all, at the same time as we found that one genetic change, we realized that there were adults who had been, you know, had normal development their whole lives, were in their 40s and 50s, and then were developing problems that were in some ways reminiscent of the problems we saw in the little kids. They developed speech problems, they developed motor problems, but still they had this long period of typical development. And then we also found that there were patients who didn't have the the typical features of basal ganglia and cerebellar atrophy, but who did have hypomyelination. And we recognized that within that group of patients, there were some patients who were very severe and some patients who were very mild. So milder and more severe than the original described children. And for a while, there was a spat of like letters to the editor, which is how civilized scientists fight with one another. They write letters to the editor, and the other one writes another letter to the editor. And there was this spat of people saying, no, if you don't have basal ganglia atrophy, you're milder than the classic HABC patients. And then the other ones would go, no, you're more severe. And they were both right. They were just looking at different patients. And now we've evolved even further that we know that there are children who have, unlike the adults who have long-term intact development, children who might have neurobehavioral deficits, things like autism, but who really have little or no myelin differences early on. And it's too early for us to know how those children are going to evolve over time. Are they going to develop myelin differences? Are they only going to develop some of the basal ganglia differences? We just don't know, and we'll have to continue that journey with those families to really understand what the entire scope of the disease is. So it's also important to know that part of the reason why we're seeing all these patients with all of these different presentations is because TUBB4A is probably one of the most common leukodystrophies, not as common as ALD or other disorders you might be hearing about in the meetings this week. But within the hypomyelination leukodystrophies, we think about on par with 4H, which is here, right? And 4H here is probably a little bit underrepresented because at the time we only knew about 3 of the 5 genes that we now cause that disease. But so we think it's one of the more common. It's, for example, more common than PMD and more common than some other leukodystrophies we've known about for a much, much longer time. And I suspect as we continue to expand this understanding of spectrum into children even without myelin differences, I think we're going to see even that population grow even more. Again, for those of you who are not familiar with TUBB4A, TUBB4A is one component of a tubulin. So tubulins are important to creating the structure and the highways of your cell. It's as if you were messing with something that was the transportation office of the cell, right? And there are lots of different tubulins, different cells have different tubulins, and also the tubulins form together, so they form with the alpha tubulin. And there's really a surprising amount we still don't understand about what these very important components in the cell do. But what we do understand is that they combine together into 2 little pieces, and they make these microtubules, right? And for those of you who have family to whom you want to explain what TUBB4A does, there's a really nice movie called Inner Life of the Cell, and you can actually see how people people visualize those tubulins coming together into microtubules, and it's really as if you, instead of having the roads that are there all the time, it's as if it was constantly a roadworks project. Like literally, you want to go to the grocery store, in front of your car they're making the highway, and behind you they're picking it up, and then you need to go to the mall later, and they need to put down a new road. And the cell is doing that all the time, and we think that part of the reason why oligodendrocytes and neurons are so impacted is because these cells need to do a lot of work. Some cells are pretty quiet, and other cells like oligodendrocytes, which make myelin, need to move proteins and fats and things around the cell all the time in order to do the important work they do in the cell. And so we think probably at least a significant component of TUBB4A is because things are not getting what's called trafficked in the cell in a proper way. And we're trying to understand exactly how, I'll go back for a second, exactly how different mutations might impact the way the 2 pieces of the tubulins fit together, how they impact how things that the cars that sort of ride on those highways stick to the highways, right? And I think probably the reason why, thinking back to, why does one patient with one TUBB4A mutation present in early childhood and one patient with a TUBB4A mutation present in adulthood? I think it's probably because those different mutations differentially impact how the things stick together and how things stick to the microtubules. All right. Any questions about that? Okay. So we are also lucky in the TUBB4A space, because we have to take our the good things about the tools we have, in that it's pretty easy to make mice and to make rodents where the mice, if you change the gene TUBB4A in those mice, you can get something that looks a little bit like the human disease. And the reason why that's good, it's not good for the baby mice, obviously, but the reason why that's good for us and for our field is that we can then try to figure out how, understanding how the mutations cause problems in the cells and understanding how we can work to reverse them, right? So those are great tools. So we at CHOP have made now several mice. I'm going to talk to you about the one where we sort of already know a lot. We picked the most common mutation to start with in the beginning for obvious reasons, but we've now also made mice with several of the other mutations, and I can tell you that they continue to do a good job sort of demonstrating what the disease is. So importantly, we can measure, and we treated these mice. I'm a doctor, I'm not a PhD, right? So I treated those mice in the same way we might think about a clinical trial. We developed outcomes, much like we're going to talk about in a minute, we developed for patients, and we really quantified them and figured out, exactly how sick the mice get, right? Exactly how they move around, and exactly what their brains look like under the microscope, right? So this shows that, I'm going to try to figure out, okay. This shows, how we can show that the mice get so sick early on that they can't feed themselves and can't take care of themselves anymore, right? And this shows how the mice are able to move around. And then this shows that in the brains of the mice, we can show both that there's less myelin, right? And that there's cell death in the cerebellum, all things that we think are probably going on in our patients as well, right? And importantly, one thing that we learned from these mice is the more mutant tubulin you have, the more sick you are. So this is a mouse with regular tubulin. This is a mouse with no TUBB4A at all, and it's really fine, right? We don't know yet if having no TUBB4A at all is fine for humans, but for the mice, it seems fine. And then this is a mouse with one normal copy and one sick copy. This is a mouse with just a sick copy and no other copy. And then this is a mouse with 2 sick copies of the gene, right? And you can see that it gets worse the more mutant tubulin you have. And we think it's because the more mutant and the less normal tubulin you have, TUBB4A, the more it impairs the cell's function. So this opens up all kinds of treatment opportunities, because it means if you can decrease, even to some degree, the mutant TUBB4A, you'll probably be able to make function in the cell work better. We started trying to develop something called antisense, right? And antisense is one of a now series of tools we have in a lab to reduce the amount of a mutant gene that is expressed, right? The way it works is that when you are making a gene into a protein, the cell copies over the gene into something called RNA, which we've all heard a lot more about because of COVID, and then you turn that RNA into a protein. So antisense impairs the ability of the cell to turn that RNA into a protein by connecting with it and preventing it from entering sort of the protein translation machinery, and so you don't end up with the mutant protein. And it's never an all or none, it's always a degree, right? But the hope is that you decrease the amount of mutant protein that the cell has. So just like we were able to do genetically in the mice, right, by just reducing the amount of mutant TUBB4A that there was, or increasing it, we're able to do that also with the antisense. And what we can show, right, is that by doing that, even just one administration, we're able to show that we can reduce the amount of loss of cells, and reduce, I'll go back actually, sorry, and increase the amount reduce the amount of mutant TUBB4A made. Unfortunately, it's important to know that we don't think the antisense, at least in our mouse models, and at least for this one, get everywhere, right? So, these things are all, especially in the beginning of trials, are all going to be about gradations. We might not treat all the cells that are made sick by TUBB4A, and we might not treat them all perfectly effectively. But even just by getting into the oligodendrocytes, right, we're able to show that once we treat them, we're able to make the myelin. So the blue here is myelin, right? So you can see it's very dark blue, right, in the wild-type mice, and also if you treat the healthy mice with antisense. When you don't treat the mice, you lose the myelin, but when you do treat the mice, you actually can preserve myelination. So that was very encouraging, and we were also able to show through all kinds of other tests, looking at how effectively signal gets through the nerves, right, and looking at how effectively sort of those signal go through and how many cells were made, that we were preserving myelination. Yeah.
So in that last slide, you said preserving myelination. When you're further along in progression, can it be rejuvenated?
So it's a great question. So these mice were all treated very, very early on, right? So in order to answer that question, you'd have to do the experiments where you took mice who were later on treated and see how they did. And these experiments are actually difficult and very time consuming, so we haven't had the resources and the funds to do that yet. We've applied to the NIH to do it, but we haven't had them. But at least theoretically, the good thing about oligodendrocytes, and from what we understand now about what's happening in TUBB4A, is that oligodendrocytes make new oligodendrocytes all the time, right? So like if you have MS, you can remyelinate yourself. You're remyelinating from that pool of what's called OPCs, oligodendrocyte progenitor cells. And what we understand about HABC now from our experiments is that in TUBB4A, the OPCs make lots of OPCs, but they fail to grow up into mature myelinating cells. So presuming that at least for a time in HABC, you still have those OPCs, the cell is still trying to overcome that deficit and make mature myelinating cells, presumably it would work later as well. So it's important to know that this trial was really only one application, right? So we only gave it once. So we know that the effect of the antisense lasts about 90 days, right? So we know that without treatment, right, the mice, we lose the mice by about 3 months of age. And you can see here that the mice do well for a time, right? And then at about the same amount of time as you start losing the effect of the antisense, they do the same thing as the mice who are untreated. So we know it maintains things, and then if you stop treating, obviously, you have progressive problems, right? But we were able to show, in the same way we had showed that the mice without treatment were sick, that we increased survival of the mice, even just with one treatment, that we preserved their myelination, right? And that overall, we made them better. Now, because these are mice, and we worried, well, if maybe this doesn't work in humans, maybe something's different about humans, we also did something in what's called iPSCs. So we took blood cells, and you can actually convince blood cells to change their mind about being blood cells and turn them into brain cells, right? It's a process that takes months and months and lots of application of something called growth factors in very controlled environments. You wouldn't be able to do it in a person, but you can do it in a dish where you can sort of control everything. So we were able to make cells that behave like myelinating cells and that they express the same kind of proteins, they express the same kind of characteristics. And with, I'll go back for a second, and with, if you have wild type cells, they make all these nice arborizations, right? The mutant ones just don't make the same things. They don't sort of stretch out nice ways, presumably with the goal of myelinating, and they don't make the same proteins. But when we were able to treat them, I'm mixing up my cursor and my front button. When we were able to treat them, we were able to get them to look much more, right, like normal myelinating cells. So that felt very hopeful to us. We were able to test also that what I was talking about, that phase where they sort of like fail to grow up, right? You don't want your myelin-making cells to be eternal children and teenagers, right? You want them to take responsibility for their world and grow up. And here we were able to show that they were more likely to do that. This is a complex tool where you measure sort of how far the little arborizations are reaching and how much they interconnect, and we were able to show that they largely normalized. So this is sort of a first proof of principle, obviously very different than putting something in a human, that again, the same principles for oligodendrocytes should work for people as well. So once we knew that, you know, we are not a pharmaceutical company, right? We're just a bunch of doctors and scientists. So we realized, though, that the pharmaceutical companies were going to need help in preparing for clinical trials. So in parallel to the efforts at coming up with proof of principle for therapies, we also started looking at how we could get ready for clinical trials. So now I'm going to pivot to really talking about that and what that looks like. And in order to do a clinical trial effectively, you need to understand what groups of patients you're treating. Remember, we talked about how TUBB4A was much more varied than we thought in the beginning, and also how you're going to measure change over time. So we really started focusing on thinking about those kinds of things. And this is where all our families stepped in, because many of you in this room have come to CHOP, have participated virtually or in person, right, in assessments, and allowed us to really map that out and understand it much better than we did. And this is part of a much bigger effort that's funded by the NIH, where we're really looking at trying to improve clinical trial readiness for a lot of leukodystrophies, and includes people who are statisticians and data scientists and who, you know, have never taken care of a patient in their lives but are just as committed as I am to making sure that we understand them, and administrative teams, including Omar Sherbini and Constance Vigneault, who some of you may have interacted with in enrolling in research. And the FDA has recently, in the last 4 years, provided some much clearer guidance for rare disease and for how to develop clinical trials. And really their message is patient first, patient foremost, right? So where we need to start is what is meaningful for families. So some of you may have participated in these efforts, but what we did is ask the families, what in your daily life is most important to you? What's hardest for you, right? And started thinking about, through these standardized scales, sort of what was meaningful. And people said, you know, it's important to communicate and be understood. It's important to be able to swallow. It's important to be able to move around. But the things that we as doctors might have thought were the most important might not be what's most important to families. And that helps us tailor picking outcomes that actually measure things that are important to families. And one of the things we really also needed to understand is within this broad variety of ways patient presented, we need to understand how can we develop tools to say, okay, this particular patient belongs to this group and this particular patient belongs to that group. So when you are doing a clinical trial, you're not comparing an adult who wasn't going to develop symptoms so much older to a baby who has symptoms very early on, because obviously if you're trying to do studies, that might really mess up your interpretation of the study if you're comparing apples to oranges, right? So we enrolled about 200 subjects, right? And those subjects came from a broad range of places and a broad range of, and had a broad range of abilities, and and a broad range of mutations, right? So we have a large number of different mutations, although the original TUBB4A mutation was still, like the D249N was still the most common, right? But we're starting to see other groups emerge with more frequent mutations as well. And we really started thinking about how could we clarify groups. And what we realized is that looking at early developmental milestones, whether or not you had head control, whether or not you could learn to sit, right, really helped us stratify patients within different groups, right? And we also realized that the common mutation sort of had its own phenotype, right? So if you put all genotypes together, right, this is developmental milestones, right? And this is P50 is half the kids gain the skill, right? So the light blue is half the kids gain the skill. You can see that in patients who don't have the common mutation and who present before 12 months of life, and I'll talk more about some specific features, who present as babies, what we call early infantile, really the number of patients who gain really complex skills, like walking up steps and talking, is much smaller than in patients who present after 12 months, where you're hitting more than 90 and 95% of patients who gain certain skills. So we started to see sort of an emergence of different groups. And also, the same was true when we talked about people who lost skills. We were also starting to see different groups emerge. And so where we ended up is really thinking, and this is an example of sort of how we can look at developmental milestones and how much people are gaining and losing them. And so these are statistical tools that we used to come up with ways that we can measure over time how people are doing, right? And what other complications they might have in the future, like needing a G-tube, like having seizures, right? Or like having orthopedic complications. I know that I'm a little bit too slow, so I'm moving along, but I'm happy to come back to some of the slides for questions. And really, we came up with these features. If you were not sitting by 9 months, right, you were more likely what we call early infantile, and if you were sitting by 9 months, you were going to be late infantile. And we know that the D249N mutation is almost always late infantile, and there are a couple other mutations that are more likely to be late. And then there were other mutations where almost all patients ended up early infantile. So we started being able to say, when somebody was coming to us early on with more certainty, you're more likely to be late infantile, or you're more likely to be early infantile. And so now we have, at least 3 large groups, right? Early infantile onset. Most of these patients have profound low tone and have really a hard time gaining developmental milestones, and will continue to have a hard time throughout their life, right? And then we have late infantile onset. Most of these children sit or walk well and get at least single words, but then may lose them over time. And in particular, we know that if you have the common mutation, you're more likely to actually struggle a little bit earlier than some of the other genes. And then we also know that there's a juvenile or later, and a lot of these patients start out not with motor features, but more features like autism and developmental differences. And so now going into clinical trials, this can help groups actually pick a specific group and designate what's called inclusion criteria. So they know that in their clinical trial, they're not comparing apples to orange, but apples to apples when they're trying to add in the nuance of then adding in a medication. So that sort of helps us do the apples to orange piece, but now we still have to figure out how the clinical trial groups are going to measure outcome. And here's where we went back to that information that the patients had told us to help them, to help us understand how we could measure, right, according to NIH, FDA standards, how we could measure improvement. And this is a large effort going on where we're still working on it, right? But we now have been lucky enough to collect dozens of assessments, right, to be able to show how patients perform over time, and in the hopes to be able to show to the FDA these tools accurately measure function in these patients. These tools reflect function that is important to families, and therefore if clinical trials can demonstrate a change in that type of function, it'll be measuring a change in function that's important and relevant to families. And then I think the last thing that I wanted to quickly talk about is just some management issues for TUBB4A. I think it's really important while we all wait for therapies and things that we can do to help change the trajectory of these diseases, also to be careful about how we manage the now. Right? And a couple things as we've sort of looked at large groups of patients, or at least large for rare disease, is we know that children need audiovisual support in school because their eye movements may be different, and they may also be processing visual things differently because we know from the mice that the signals that get to the brain are different. We know that a higher percentage of patients than expected may have cardiac differences, and so we recommend that people see at least once in their life a cardiologist. We know that our patients really need the help of a physiatrist and sometimes tone management clinics to optimize control of tone because their tone is very problematic, and that can lead to problems with scoliosis and joint dislocations, and so you really also need to make friends with a good orthopedic surgeon. We know that we want to make sure that kids don't lose weight and nutritional status while we're waiting for better therapies, and so we recommend that people get early access to feeding evaluations, and if needed, a feeding tube, and not a feeding tube when you're already really failing to thrive, but early on in your course so you can support your growth and nutrition. And then we know we need to be really careful about screening for things like seizures, supporting mobility for problems like ataxia, and screening for hearing loss as well. And then this is our clinical team who shows up every Thursday to see patients. This is a day where we're all wearing ULF t-shirts, which is why I put the photo here. And there are a lot of committed people at CHOP, but also around the country, who focus on caring for children with TUBB4A. And our team includes nurses, social workers, a lot of occupational therapists and physical therapists, speech therapists, doctors, a physiatrist, a complex care pediatrician, and neurologist. You'll see here Dr. Adang, who wears zebra clothes frequently to clinic, and these people all work together to make sure that we steward these diseases appropriately in the now and hopefully in the future for clinical trials. And I'm happy to take any questions.
We're discovering that we're having a lot of overlapping conditions, and I don't know which leukodystrophy and which is just like other complications. And some doctors are very dismissive on some things, like he has a gene defect in his heart, and they're like, oh, it's no big deal. But however, I came across another doctor who says it's going to manifest during puberty, and you really do need to watch this. I didn't even know leukodystrophy could cause cardiac abnormalities. So I don't even know if that's a separate issue or conjoined, or if we should be watching this, then I'm not getting clear direction on some of this.
We can talk offline because I don't want to make you share your medical history if you don't want to, but not all leukodystrophies cause cardiac differences, and we don't understand why in TUBB4A there are more children with cardiac differences than the general population, because at least theoretically TUBB4A is not expressed in the heart, so don't ask me why, I don't understand it. But we just know from observing families that yes, that probably is related depending on what kind of cardiac difference. And so I think that's part of why it's important to come together as a community and share data, because only by, because about 1 or 2 kids out of 100 will have a cardiac difference. So it's only by sharing large groups of patients that we start seeing emerge patterns that help us understand what's connected and what's not connected.
So on that same front, I know there are families across the nation that don't have the ability to come to CHOP to be assessed. Is the other network hospitals, will they do the same degree of review and care oversight, or at least initial?
So we can't impose standards of care on other doctors. It doesn't work like that. But we have published our recommended standard monitoring as an appendix to our recent paper that outlines the different apples and oranges conversation I was talking about. So for anybody who needs to come to an appointment with that sort of guidebook, it's findable, and you can contact us and we can get you a copy, and then you can just go saying, look, this was peer reviewed, it's published, this is what we need you to do. That's our best tool for helping establish standards of care is publication.
With this autism on the rise across everywhere, do you see any heightened levels of autism in some of the TUBB4A? Is it trickier to navigate because he can't, say, hey, my leg hurts today?
Yeah.
Do you see any of that trickling into some of your research, or is it completely separate?
It depends on, so some people say that, so it used to be that you were only diagnosed with autism if you had no other neurologic explanation, right? So it depends a little bit on how you define autism. Now that genetic testing is so much more available, people are recognizing, of course, autism has lots of causes and lots of genetic causes. So people are more flexible with their terminology. But certainly there are a subset of more mildly affected children who present with features consistent with autism and who probably would benefit from the same kind of behavioral management as autism.
Anybody else?
Okay.
Thank you. Thank you very much, Dr. Vanderver. Appreciate it. And I know she's got to run and get on a plane. So, all right. I'm going to ask Uwe Meya from Synaptix Bio to come up. This would be a good segue into what he can share with us relative to the pending ASO clinical trial with Synaptix Bio.
Well, good morning, and thank you for inviting me here. It's my pleasure to talk to you. I have no slides, but I wanted to use most of the time really to address questions that you may have. But before I do that, I give you just a brief introduction. If you are interested in Synaptix Bio, you can also visit our website, synaptixbio.com, where you find all the information. But in brief, I'm Chief Medical Officer of the company, and the company has exactly 2 employees, which is myself and the CEO. So anything else is done with contracting to external experts and consultants. We are located near Oxford. That sounds nice. But actually it is just a postal address. We have an office that is half the size of this room and consists of 1 table and a few chairs, that's all. We are not located in Oxford, which would be a nice touristic spot, but outside next to us is a cyclotron, and on the other side is, in spite of Brexit, the European Space Agency. That means I myself live in Switzerland, which is a nicer place than that business park, and work mostly home office based. So having said that, we, as a company, we have the paradigm, we want to develop a drug for the treatment of HABC and eventually other leukodystrophies in a way that it is finally a fully commercializable product. So that is brought to the market, prescribed by physicians, and treated according to the approved indication. And that means we have to comply with a bulk of regulatory requirements before we can even treat a single patient. So we do not have the paradigm of trying to take shortcuts and treat maybe 1 or 2 patients and see what happens. But we are committed to full development and later on to full commercialization. And in that respect, we enjoy very close collaboration with CHOP and Dr. Vanderver and her team throughout the process, so really from the test tube to patient data. We have to comply with something that is called CMC, that is chemistry, manufacturing, and control. So to make sure that the molecule that we synthesize is really what it is, there are no impurities, that it is stable, that it is not toxic or any of its metabolites are not toxic, and that it is able to be put into like a vial for administration, that it is stable, that it doesn't have any toxic degradation. Second is we have to show in animal experiments that it is reaching the target and doing what it is intended to do. So you have seen all the experimental slides from Dr. Vanderver. Essentially, we have gone through all that process. So we have shown that in the test tube, our molecules are able to reduce the toxic TUBB4A. We put it in various animal species. Also, we were able to show what you have seen here from Dr. Vanderver's slides, for example, that it improves survival, that improves motor skills. You have seen the rotor rod where mice sit on a rotating rod, and you see, can they cling to the rod or do they fall down? So it is experiments like that. All that has gone very well. We had the luxury actually to select one lead compound now between several that we had. So several looked suitable, so it was a nice problem to have. And we are currently in what is called IND enabling studies. That means it's really the last stage when you have selected your suitable compound that fulfills all the requirements and put it into the formal experiments that allow you to treat patients with it. The biggest thing among it is toxicology. We have to test it in more than one animal species, and we have to test it for a certain length to be able to treat patients for extended time. So long-term toxicology as well. That is currently in preparation. We, as the clinical team that I have with external help, we are going through the process of regulatory advice. As this indication is a so-called orphan disease, a rare disease, we have the privilege of more frequent interactions with regulatory authorities than for well-established big indications. We have had several meetings with the British MHRA, which is the regulatory body there. We have had several interactions with the FDA. And the process is that you tell them where you are, you tell them what you have in mind, and you ask them, do you agree, yes or no? And these questions or interactions so far for the clinical preparations have focused on 3 topics. Topic 1 is patient selection criteria. So who is going to go into our clinical studies? The second is what endpoints are we going to use? So what efficacy parameters are considered valid by the regulators to approve it finally? Regulators say at the end of the day they want to see a parameter or a set of parameters that tells them, is the patient doing better in everyday life than an untreated patient? And the last, that is also a very difficult and controversial topic, is what control are we going to use? So the FDA wants to show that our drug works compared to a patient group that is not treated. The big elephant in the room here is the placebo. Do we need a placebo control? I have seen some remarks circulating in social media, and I wanted to address them. So the first is about patient selection. I saw a remark like, we are targeting pre-symptomatic patients. This is not the case. It's impossible. We do not recruit patients, or are not intending to recruit patients based on a specific mutation. We have seen from Dr. Vanderver there are more frequent and less frequent ones. There is some hint that this or that mutation may be associated with a more severe or less severe cause, but it is not justifiable to exclude patients per se on a given mutation. So that's one thing. What we are planning to do, and that addresses the next remark that I've seen, are we years away from clinical trials? We are not years away. So the plan and the hope is that we can start next year with patients. We want to start with a clearly defined and narrow patient population. In terms of Dr. Vanderver's remarks, these will be the so-called late infantile patients, so those who have reached initial motor milestones but have started to decline. So they are symptomatic, not pre-symptomatic. and those who have at least some ambulation left, so who can at least walk with maybe assistance, bilateral arm support, etc. So that will be the very first cohort, and we are targeting the UK for that. It has several reasons. One is we are partly government funded, so there's an expectation that we do work in the UK, but even more, there is a network of excellent experts in London, Birmingham, Manchester, who can deal with this patient population. But as we gain confidence in what is a safe dose, how high can we go, what is the highest well-tolerated dose that we give as a maintenance treatment to patients. So as soon as we gain confidence, we want to rapidly expand recruitment into other territories. And then the USA is clearly number 2 where we want to go. And secondly, also broaden the indication. That means the regulators have already advised us to consider very rapidly patients who have lost ambulation and or those so-called early infantile who have never reached ambulation. So that is the plan that we have, and hope, as I said, we can start next year to roll it out. So it will be gradually, step by step, also making sure that we have an adequate risk-benefit risk-benefit ratio. Then there's another big question, as I said, placebo control. So no one will like it. I have done in my previous jobs placebo-controlled studies, also in progressive diseases. Some patients said, well, if it is only for a defined period of time, and maybe if it is a so-called 2-to-1 randomization, that means if I have a 2-to-1 chance of getting active drug and not placebo, and if at the end of the day I will get active treatment, I will do it. Yeah. Particularly the FDA has a very strong bias towards placebo control, but we are currently discussing with them and working with them. Is there anything that could substitute for that placebo control? For example, could the natural history cohort that Dr. Vanderver has recruited, could that substitute for a control group? So could that represent a group of patients where we can compare our treated patients with? The FDA is very cautious about that. So first of all, they want to see these data in every detail. They want to make sure that we have no selection bias. So we cannot go to that natural history group and just pick those patients out of it that fits well into our paradigm. Yeah, but we have to make a clearly methodological selection. There are also some interesting new biostatistical approaches. I don't want to go into detail with that, but there are biostatisticians that can model from existing data and sort of forecast what untreated patients, how they will behave in the future, and we can compare to that. So this is broadly where we are. A lot of work, and of course, I've been more than a year with the company, and now preparing. There's also, I have a desire now to see, let's get started. Yeah. And last thing is also like your input, how do we administer the drug? So it has to be administered into the cerebrospinal fluid to reach its target. There are several potential methods. One is called intrathecal, so you put a needle in the spine, injected. With that you can also draw a sample of cerebrospinal fluid and see what the drug, how the drug is metabolized. That has the disadvantage, like in very young patients, it will likely require sedation to keep still for a while. You can't do that too often. Another method would be like a depot with a port, so you can implant a port and insert a cannula into the brain even. That has pros and cons. Of course, it doesn't require repeated lumbar punctures, but on the other hand, it is, of course, it's a small neurosurgical procedure. But once the port is implanted, you can just inject through the skin and draw samples, so it doesn't require repeated lumbar punctures. There are issues around hygiene, risk of infection, displacement. So that's another point that we are working on. But as I said, we desire your input. Last thing is efficacy endpoints. As I said, at the end of the day, the FDA wants to see, do you delay the progression? Can the patient sit, stand, walk longer than as if they were untreated? What is their quality of life? Is there any impact? But these parameters are likely to change very, very slowly. So it takes a long time to see that effect. So we are also testing so-called unapproved, from the FDA, unapproved devices. So it's like wearables. It's like a wristband that you attached, and it can measure your movements. We are also working with a company in Melbourne that assesses speech and language. So it records language in a day-to-day interaction with a patient, and they have a very sophisticated system to analyze that, so like the pitch, the frequency, the length, and whatnot. Again, when you present it to the regulators, the FDA says it's an unknown animal to them. What is it? What does it mean? That is the first question. But again, we are inviting patients also to work with us to test it right now. It's at no cost to you or to us. So that's another idea that we are currently pondering. Yeah. Okay, so that's what I can say. So there's much that I cannot say. Exactly what and when and where do we start, but at least you see we are working very hard to make it happen.
Thank you, Uwe. This is great. And we'll be reaching out to the families on some of the mechanism equipment that you've suggested might be of help. Does anybody have any questions for Uwe? Now's your time.
I already asked a few questions. I made a mistake of posting publicly on Facebook that your website said 2028 was the hope of trying the clinical trials, and I did misspeak because you had said you're trying to do it sooner. When you do have those dates or no, I mean, you are going to post those publicly, so we're anticipating those updates.
I will be here at these meetings regularly. I will be there at other conferences. You can always check our website where we make announcements. I think, so shame on me, I didn't check our website, but I think we announced that we have a final candidate selected for the formal non-clinical investigations. But so apologies, as an employee, you are the last one to check your own website. Yeah, so but they will appear there regularly, and we post them. Yeah, there was also another remark that I saw on social media that only patients will be eligible who live close by a site. Again, that is not the case. So in the UK, as I said, we have London, Birmingham, Manchester very likely. In the US, we have a couple of expert sites, of course, CHOP in Philadelphia, but there's also Boston, Baltimore, Salt Lake City, Stanford. Travel shouldn't be a big issue. If we do so, there are professional travel agencies for patients who can assist in travel. It has to be reasonable. I mean, I've worked in other clinical studies before. We were provided that, so no 5-star hotels, no business class flights, but a decent amount of support is also accepted by IRBs.
My question is how many people, or you can't discuss that yet, potentially, but is there a general number of patients that would be asked to enroll?
The eligible patients, as far as we know, is not a big number. So that means we do not have a top cap. We will start with few patients in the UK, like 6 or 8 or so, but then expand, just see as many as we can get.
So this might be too early, but I heard something about how it's going to be administered. You talked about a port. It reminds me a bit of a baclofen pump type thing. Would this be something down the line where it would be administered, if successful, in that way?
I mean, that's a long shot, of course. For the clinical studies, we aim at one single way of administration. So you could induce a bias or unnecessary variability if in one patient's injected into the spine, in another patient's injected straight into the ventricles of the brain. So we want to make it all the same. And also the toxicology studies that we are doing. So there's one species that is monkeys. We want to make sure that we administered already in the monkeys in the same way as we plan to do in the patients. What the final way of administration will be, I cannot tell you that. I mean, obviously as convenient as possible and as safe as possible.
And I do have a question for some of the parents with older children. A lot of our kids have intervention for spinal fusion, scoliosis. I know for a fact I've looked into it for my child. And how are the risks different for the younger versus the older kid that have been more involved?
I mean, that's something that we have to bear in mind. So like a spinal fusion, is that prohibitive for intrathecal administration? Again, I have no solution yet, but we are also discussing it with neurosurgeons who have extensive experience in intracerebroventricular administration.
Go ahead. My son is young, obviously, and then I see Eloise, who is older. In some of the hopes was to reverse some of these things, right? You can reverse contracture, you can't reverse fusions. So is there just a range of things they're hoping to reverse, or do they know what's being reversed in the mice?
Well, we could replicate with our molecule what Dr. Vanderver showed, that we can restore myelination, so which is of course the first step to make the oligodendrocytes functional and help support the neurons. That is what we can say, and what we could show in the behavioral experiments, as I said, this rotor rod, that the mice have a preserved or better capacity to move. So, and that is hopefully what we can replicate in patients.
Thank you. And then an online question: how long after you've started the trial in the UK do you think you might be able to bring it over here? And then are there other alternatives with expanded use that the FDA may allow if we can tap into what you've done in the UK?
First question is how fast we can get on in the UK depends on the recruitment speed. I mean, apart from a whole bunch of administrative procedures, we have to get the green light from MHRA, we have to get green lights from ethical committees, we have to conclude hospital agreements, so hospital, they will charge us for every blood draw and for every x-ray, so it is not to be neglected, so that is a lot of paperwork, but then it's dictated by the speed of recruitment, how fast we can get the patients. So we hope to have that completed in the UK within 1 year. Obviously also we need funding, we need more money to expand the program, and if the data look good, I think there is, we can be optimistic about the funding, and then as fast as we can expand into other countries. So we are not tied to saying we will only finish in the UK and then see what happens, but if we get funding and good data, we can accelerate.
Thank you, Uwe. This has been very helpful. And I will say the transparency and the information is extraordinary for our families to understand where we're at. Thank you very much. Dr. Waldman, please. Thank you. We're mixing up the schedule a little bit just because we're a little bit behind, so I've switched things around, but you're cute to go next. Thank you very much.
Well, I'm delighted to be here, and I just want to start by saying, so I'm Dr. Waldman. I'm at Children's Hospital of Philadelphia. I've had the pleasure of partnering with Dr. Vanderver and so many of other team members who are in the room and hopefully online and throughout the world. So I have the job of typically bringing these treatments to clinical trial. So we just heard, how do I get my slides? We just heard about hope, right, from Dr. Vanderver and Synaptix Bio, and I think we should all be so grateful that Synaptix Bio has chosen HABC to bring to clinical trial. Just, hi, buddy. Okay. Good. Yeah. I do have slides. I don't know if they're coming up. Green button. Oh, there we go. Okay. So we just heard all about the hope of bringing a treatment to this disease. However, with that hope comes a lot of other emotions, right? So there is sometimes fear of a new treatment. There's sometimes jealousy of what might be brought to an opportunity for one patient versus maybe not an opportunity for other patients. So there's a lot of things that go into these clinical trials. But I'm going to try to walk you through, as parents and as community members, the realities of clinical trials. I'm not going to spend a lot of time on this, especially because we're behind, because I want to get to the meat of the presentation. But there's many types of studies, the natural history studies, the registries, clinical trials, which is what we're going to talk a lot about today. And I just will mention, because there was just this question about expanded access or compassionate use, right? I will just start by saying those mechanisms in the United States do not come into play until we have safety and efficacy data from a treatment. So if something is tested in the UK, and you can jump in here if you feel differently, it is not going to be able to go through the compassionate use mechanism with the FDA because there's no safety or efficacy data yet, right? Once you have safety and efficacy data, and a lot of times data that's been collected in the US, then you can go down to compassionate expanded access use with a drug. When it's still in the experimental phases, it's very hard to do that. So I don't think that that's going to be an option when this is just being started in the UK. I really want to take families through what it's like to participate in a clinical trial. And I'm going to actually, where's Michele? Do you want me to skip through a lot of this, what a natural history study is? Okay. We're going to skip through these types of things and go really into the clinical trials, and we can always go back if people have questions. So as we were just talking about, clinical trials are the hallmark of evaluating a new therapy, so we're really excited about this ASO, this antisense treatment from Synaptix Bio, but it's new, it's investigational, and we need to study it. The two goals, is it safe, and does it work in humans? Right? We go through, and our partners go through this extensive work in animals, but now we have to figure out, do those animals translate to humans? And there are some unique aspects that you've already heard about. There's randomization, which is the unknown assignment. No one picks, but you get assigned a treatment or no treatment or placebo. We talked a little bit a few minutes ago about those placebos. I actually am in favor of placebo-controlled trials. I know as a family, everyone wants to get the drug right away, but that really is the hallmark of trying to determine if something works or not. So these are the things that will come into a clinical trial design. I'm going to go very quickly through the different phases of a clinical trial. There are phase 1, phase 2, and phase 3 trials. Typically, you don't get approved by the FDA until you've done a phase 3 trial. So you have the beginning phase 1, phase 2, which can be sometimes combined, but the phase 3 is the pivotal trial, which goes to the FDA. And then we'll skip over some of this in terms of how we evaluate these trials, but the primary endpoint has to be clinically meaningful to the patient, and it is sometimes very difficult to find clinical endpoints that are applicable to many different patients of different ages and different abilities, and so those are challenging as well. We talked a little bit this morning about how would you measure if a drug is working. There was this statement just a few minutes ago about reversing neurologic symptoms, and you correctly said you can't reverse a contracture, you can't reverse a scoliosis surgery. For the most part, I think we have to go into these trials with the idea that we're not going to be able to reverse symptoms, that we may not be able to allow a patient to gain new skills, that there might be a plateau or a stability as opposed to a reversal or a gain of new skills. But these are all of the questions that are being debated right now in the HABC community. How are we going to measure if the drug works? Who are we going to be able to study the drug on? I know some of you might be disappointed to hear that the earlier stages of disease might be targeted first. I was going to give an example of blood pressure. I wanted to stay something away from anything neurologic. But if your blood pressure is controlled, we don't know, for example, if you're testing a blood pressure drug, if that drug is doing anything or not. So you need to try to enroll into these trials patients that have symptoms that we might be able to show change a little easier. And that's why sometimes younger patients might be enrolled versus older patients. And again, the trial's still being designed. I'm just giving examples. In other areas, sometimes we might enroll adults because we want to make sure that it's safe in adults before giving it to children. So there's a lot of things to be considered in terms of the trial design. As you heard, it takes a long time to develop these trials. There's a lot of thought process and going on to things now while we're trying to figure out the safety and efficacy in the animal models. These conversations are happening now. There are ethical standards and strict rules need to be followed with these clinical trials, right? So it's not always the physicians that are seeing the patients that then have to go back and design the trials. We advise the companies, but then they come up with their ideas, and then there is no wiggle room. If they say this is the age parameter, then we can't say, well, I'm just aging out or I'm aging in. It's a very strict process and very strict rules that need to be defined. There's a very specific and detailed protocol that cannot be changed or wavered. If you waver from the protocol as an investigator in a clinical trial, you get what's called a protocol deviation, which gets reported to the FDA and to the pharmaceutical company, right? So if we say, well, something, we don't want to do it this way, we're going to do it this way, it's a protocol deviation and that gets reported, right? So there's a lot of very strict rules that need to be followed. And so, and we already talked a little bit about the placebo. But I want to explain a little bit more what I mean about these strict protocols and stuff in a few minutes. I just want to remind you of these points in clinical trials. Clinical trials are research studies, not clinical care. So the proposed drug is unproven. There's a lot of data to support it, but it is unproven. There's no or limited safety data, right? There might be data in animals, but not in humans. There's no or limited efficacy data, now depending on when you come into the trial. If it's a phase 3, then there is some of this data already. But in a first-in-human trial, a phase 1-2, this is unproven. Clinical trials involve risks that are different from clinical care, and we're going to get into that in a few minutes. Even if the FDA or our European equivalents, even after the FDA approves a trial, it's still up to the individual institutional review boards to ensure that patients are kept safe. So we've had the experience where the FDA has said, okay, this study is approved for humans, but some of the IRBs, the institutional review boards, have said, you can't give this drug to children until you give it to adults. Now, the advantage of this being done in the UK first, especially done in the UK on younger children, is that we will have that data for the FDA and the institutional review boards in the United States to avoid that problem where we might have to go into an adult before we could give it to a child. So as much as we sit in this room and we're all kind of feeling a little jealous that the UK colleagues might have first access to this drug, by the time it comes to the US, you will know that there's some safety data and efficacy data. Well, we might not know the efficacy data at first, but there might be some safety data before it comes to the United States. So I want families to think about things. Of course, we're all tinged with jealousy to hear that other patients might have access to it before we do, but there's also advantages and comfort for you as parents to think about, well, okay, my child might not be first, but my child also might not get the lowest dose, or my child might have the advantage of having some safety data before coming to my child. So I really want families to support each other and the community to support the patients that are in this trial, because it's really a very tough thing and a big ask. I put in red a comment that was made by a pharmaceutical company that I had never heard before, but was very eye-opening for me. The goal for any clinical trial is to enroll the fewest number of patients to show that the treatment is safe and that the treatment works. I think we all think if there are 50, I'm pulling numbers out, but if 50 patients in the world with HABC or 50 patients in the US or whatever it is, that all of those would have access to the trial. We think about like blood pressure medications and how those are thousands of patients in the trial. So we think all of our patients should be able to get into the trial. That's not how this works. Their job, Synaptix Bio job, is to try to show that their drug works with the fewest number of patients. So not every eligible patient might have access to this trial. This is key. Who is the best candidate for a clinical trial? Now, before we get into this, as you heard, there's going to be inclusion and exclusion criteria. So I'm assuming that those inclusion criteria are already met, right? Hypothetically, if there's an age range for the trial, you have to be, not hypothetically, but you have to be in that age range in order to be eligible. But once you meet all of the inclusion criteria and you don't have all the exclusion criteria, the best candidate for a clinical trial is the patient that has been medically, nutritionally, and emotionally stable prior to enrollment. This is different from oncology. A lot of our cancer trials, patients are enrolled after they have failed standard cancer drugs. Okay, that means they're some of our sickest patients that we give experimental therapies to. That is not the case here. And of course, if you feel differently about any of this, please jump in. But the best candidate is the one that is going to be stable. And sometimes that's stable for a month, sometimes that's stable for 3 months, and sometimes even longer. And when we say stable, what we mean is no medication changes, no adjustment in seizure meds, no adjustment in tone meds, no new physical, occupational, or speech therapy, right? If we're measuring ambulation and the patient just started new physical therapy, that's also a problem. So there will be rules around this, again, how many patients can, or how long you have to be stable before getting into a trial. So if your loved one is getting worse, they may not be eligible. Again, the advantage of doing this in the UK first is we might have some of that idea. Like the patient has to be stable for a month, or the patient has to be stable for 3 months, or the patient has to be stable for 6 months. So then us as physicians can help guide the community a little bit and say, okay, we need to start physical therapy now, not wait until the trial comes to the US. Now, of course, many of you already have physical therapy, but you understand the analogy and where I'm going. So this is very important. This is also just as important. The most important patient, or the best patient for a clinical trial, is the family that can comply with all the study procedures. That is what I ask my families. Are you going to be able to fulfill your contract in this trial? This is a contract between us and our patients, and of course the pharmaceutical companies. You have to be able to fulfill your job, which is to communicate regularly with the site, be at the clinical trial site for all the procedures on time and ready to go. It also might mean, we talked about spinal taps, we talked about procedures, we talked about these things. If you're getting a spinal tap a week after Thanksgiving or a week after Christmas, right, and you have to be sedated, maybe you don't do a big family gathering that year because you don't want to get sick and have to cancel the procedures. It's a huge commitment to families, but there's so much that goes into planning these visits that we really need people to be 120% in and committed to making sure that things go smoothly, right? And you might say, well, why don't we do it before Thanksgiving? Well, the protocol gives you a certain window of when things need to be done. And if that window says it's the day after Christmas, there is no wiggle room to move things around. Otherwise, you get that protocol deviation, which gets reported to the sponsor and the FDA. So for me, the most important thing is that the family that's going to answer the phone, answer those texts, answer those emails, respond with us regularly, keep track of everything. The other thing in a clinical trial, we have to write down every Tylenol, Motrin, acetaminophen, paracetamol for our European colleagues, every dose. We need those families that are going to keep meticulous records, bring them to clinic. My kid had a headache on this day, I gave Tylenol. I went to the pediatrician for allergies. They suggested I start this med. Is that okay in the clinical trial? We need people that are going to be very, very detail oriented because all of that needs to be reported, not just the stuff related to the neurologic condition or the spinal tap. So we need families, no missed appointments. Many of you will be flying to your clinical trial sites. Please come early. I mean, I don't know if people had trouble getting here. We certainly did have trouble getting into Chicago. You know, things were late, flights were delayed. Thank goodness between Philly and Chicago, there was many, many flights and we could sit on the tarmac. But if you're coming from North Dakota to Philly, which we have for clinical trials, and you have to get on multiple flights, please come a day or two early. A lot of times the pharmaceutical companies will support those extra nights in a hotel room to make sure that there's no changes in the protocol. And if we miss that time slot with anesthesia, it is very hard on short notice to make that up. So we need people to arrive early, especially the day before or 2 days before, engage with us. The last thing that I will say is the way these clinical trials work is that a lot of your travel can be reimbursed. We'll have to talk about whether that's feasible, but there might be, and I've seen this in other trials, there might be caps. For example, $250 for a hotel room, which might not be the closest hotel room to the hospital. I have to say a lot of pharmaceutical companies are very understanding and in Philadelphia let you stay at the nearest hotel to the hospital, even if it's a little more. But we sometimes have to get approval for that, right? Flights. We have to book early to get the best pricing, right? But there are additional costs. And a lot of times, flights and hotel rooms can be prearranged so that they're not coming out of pocket. But for a lot of other families, they have to put the money up and wait for reimbursement. Food, same idea. Sometimes food can be covered, but you might have to pay for it, submit the receipt, and get the reimbursement. So people need to think about financially, is that going to be feasible for them? You sometimes get a little bit of a stipend, like a $50 or $100 payment for participating in those trials. That's not going to make up for lost wages when you're missing work for 3 days. But we can't give more than that per visit because then it's coercion, right? So there is a financial cost, even though pharmaceutical companies try to limit the financial cost to families. This is just something to think about. And it goes back to that meticulous note keeping and record keeping, especially with receipts. You know, if you bought a coffee or if you bought this, you need to keep those receipts and submit them to us. And again, the last thing I will say, the schedule is determined by the protocol, not the patient, not even the physicians. We plan our visit dates 3 and 6 months out. So sometimes in the beginning, it's hard to plan, but once we know that this is the date you enter the trial, then we can plan all the visit dates and we will give those to you, but there's no wiggle room. It has to be driven by the protocol. All right, this is going to be the toughest part of the talk today. Clinical trials come with risks. And the question for each individual family is, how much risk are you willing to accept and for what benefit? We have just said that there's some neurologic symptoms that cannot be reversed, you know, and that sometimes the best thing that we can hope with the drug, that it keeps the patient stable and keeps them alive. But how much risk are you willing to accept for that? This is going to be a tough slide, but I just think it's important to walk everyone through it. Okay, not this slide, the next slide. But anyway, all right. Before I get into that, I just want to talk about, there's a couple of different things we mentioned in terms of clinical trials. There's the gene therapy, which is a one and done model. Sometimes this is placebo, sometimes this is not. In these gene therapies, you get the treatment or the placebo, and then you get the treatment, and then there's no more treatment given. It's just a lot of monitoring. I'm not going to go into that one today because we're really focused on the antisense trial. The antisense trial, you need to give these drugs over and over again. Typically, it's been every 3 months. Sometimes it can be more, sometimes it can be less, but you would have to get the therapy every 3 months, which means you have to travel to the site every 3 months, but you also in the beginning might need to travel every month for us to measure how the drug is working. So there's a lot of visits in an antisense trial. We already talked about expanded access, so I won't go back into that. All right. We talked again about the best candidate, but when you're thinking about risk, I want you to think about the what ifs when deciding if this trial is right for you. Okay? What if there is an adverse event or side effect in another patient, not your loved one, but somebody else? Are you going to feel comfortable continuing in the trial? So let's use a silly example, but just that is probably not relevant here, but just as a point. Let's say someone getting this therapy develops liver failure. Now, this is a drug that's going to be injected into the brain. Liver failure is not something that we're really worried about, but I'm just throwing it out there as an example. You're in the trial with your loved one, and you find out that somebody else has developed liver failure. Are you going to feel comfortable continuing the trial? If we know that the liver failure is related to study drug or suspected to be related to study drug, then we will have an obligation to share that with you, but there's a lot of unknowns. We might be saying to ourselves, now again, this is a silly example, but please just go with it. We might be saying to ourselves, well, we don't really think it's related, but it could be related, and we don't really know. Are you going to feel comfortable continuing in the trial? If the answer is no, then maybe clinical trials are not right for you. What if you're the one, your loved one is the one that develops liver failure, and we're sitting there saying, well, liver failure is not common after antisense drugs, so we don't think this is related. And you sit there and you say to me, well, how can it not be related? This is the only change that my child has gone through. And we said, well, we want you to keep getting the drug, and we'll see if the liver gets better or gets worse. Are you going to feel comfortable doing that? Now, I will again say, if we think that there's something that the drug is causing a side effect, there's a data review, there's safety measures in place, but a lot of times in clinical trial, it's not clear, right? And we do not expect liver failure from these drugs. So if liver failure happens, we're going to say, that's odd, that's not expected. Let's continue to give the drug and see what happens, right? So that's a scary place for families to be. If you say, I don't want that, maybe a clinical trial is not right for you. What if there's a loss? This has happened. This has happened. What if something happens to a patient? And I'm trying to use my words pretty carefully with everyone in the room. What if there's a loss? You know, and we might be saying, we don't know that it's study drug. This patient has HABC. Maybe it was just the underlying disease. Are you going to feel comfortable going through with the drug? And we're saying, well, we don't really know. Now, If there is, heaven forbid, a side effect, and we do think it's drug-related, again, there's a lot of safety measures that we go through before giving it to another patient. Sometimes there's pauses, sometimes there's other things in clinical trial, but these are the realities. These are things that I've lived through with patients and families. And it's tough. It's really tough, and it takes a lot of bravery to go through a clinical trial. And I think a lot of times families look at the families that get into the trials as the lucky ones. And I understand that mindset to say, like, you're lucky because you're in. I'm not lucky because my child's not get in. But these are the scenarios that these families have gone through. And they can't talk about it, which I'll get to in a few minutes. So if the answer to any of these questions is no, then maybe a clinical trial is not right for your family. And that's okay. This is research. This is not clinical care. Research is optional. Research involves risks. And if you say, do no harm is that tenet of medicine, if you as a family member feel do no harm, that you don't want to take a chance or experiment with your loved one, that is just as brave a decision as it is to enter this trial. And it's okay. People should not feel guilty about saying no to trials, but people should also understand that the jealousy of the patients that got into the trial, the people in a trial are often in a very tough place. Okay. So here's my advice for families. Talk to your extended family, your support systems, to really ask the question, is this right for me? If you're interested in participating, once a trial comes to the US, then let us know you're interested. Oh, let me say one more thing. Again, jump in if you disagree. Oftentimes it is not possible to go to a different country to participate in a trial, and nor should you. The health system in the US is very different from the health system in the UK. Insurance in the US is different from insurance in the UK. Clinical trials do not go through insurance. However, heaven forbid something happens and you end up in the hospital in the UK, you have no idea if that is going to be paid for by US-based insurance. And things happen. We've sent people to the emergency room. We've had to admit people in clinical trials for safety and for other concerns. It's really not that easy to go across country lines and participate. We often say you're not allowed. Now, there's exceptions sometimes, but there are serious risks with the clinical trial, and adding in some of these extra challenges is really not advised. But once it comes to the US, for example, if you're interested, you have to contact HABC, contact the pharmaceutical company, contact the site. All of the information will be listed on clinicaltrials.gov of who to contact. Tell us you're interested, right? Because of all the risks of clinical trials, we're not going to chase family members down. We're not going to say, hey, you interested? You want to participate? You want to do this? Because that's putting influence on you. So let us know that you're interested. I will say that eligibility does not guarantee a spot in the trial. Just because you're in the age range, just because you're medically stable, does not mean that you will get in. Please be patient with the study teams and the sponsors. We are all working behind the scenes diligently. It feels like no progress on your end, when I promise you, this is at the foremost of all of our activities and our goals. It just takes a long time. There's a lot of things that need to be done. You heard on the pharmaceutical side, on our side, to prepare for a clinical trial. Clinical trials often bring a roller coaster of emotions: hope, fear, jealousy, anger, frustration, fear. Again, it's a roller coaster. Study teams are under confidentiality agreements, and sponsor cannot share information that impacts the integrity of the trial. I took the slides out, but I have an example of I was able to identify by name patients in a particular leukodystrophy trial that I was never a part of, that was not even in the United States, because of social media posts. I ask my families to never post on social media. Here, I'll just go back for 1 second. Social media is problematic, in my opinion, for a couple of reasons. First of all, in this other example, there were some social media posts about these patients that I can identify by name. And the social media post went through the roller coaster of emotions, and some of the positives and negatives that the patients were experiencing. So if I'm the FDA and you're telling me the drug's working, I have all these social media posts that say that this patient's suffering. And so I'm supposed to believe your data when these social media posts say otherwise. It just gets very, very complicated. So that's number one. Number two, as exciting as this is for the community, there is that idea of the haves and the have-nots. The I got in the trial, but you didn't. You don't want to rub it in anybody else's face. It's not fair. It's not fair. We're in this together as a community. What benefits one patient, we're hoping is benefiting another patient. What happens to one patient, we hope might not happen to another patient. But there's a lot of angst that goes into these trials, and social media is not a place for it. If you feel that you want to update your family members and your friends, do it through an email, not social media. Do it through like a private, source. Do not post it on social media, and ask your family members and loved ones not to post. There should never be a post, hey, congratulations, so-and-so, you're going to CHOP to be in the clinical trial. That just identified you. Please, 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 do not post on social media. Is that a rule? No, we can't make it a rule. But this is a contract that you're signing. And we need things to be private for your own sake, right? So please, no posting on social media. All right, so just my last closing remarks, lessons learned, besides no social media posting. These are emotional roller coasters, right? And we just took you through that this morning. Dr. Vanderver starting with the hope and all the data, and Synaptix Bio telling you about all the excitement that's happening in the next few years. And then here I am saying, well, but here's the reality of this. Trials are expensive, and I'm not just talking financially, but they are financially millions of dollars. There are costs to the family. A lot of times you have to put up the money in order to be reimbursed to come to a travel site. But even more so, these are emotionally draining. There's an emotional fatigue with a lot of these trials. Please support each other. It is natural to feel jealousy of a patient that can get into a trial if you can't. But think about what I said and all of the emotional roller coasters. Getting in might be the easy part, right? As we start to test the drug and more things happen, it gets a little more complicated. So that's the end of my presentation. I'm happy to take any questions. We're very excited about the future. It is our job to make sure that families are informed. And when you participate in research, you have to be very well informed about where things could go differently than expected. So, any questions?
Thank you, Dr. Waldman. I asked Dr. Waldman to come up here and share her experience with PMD, and I said, I need you to be forthcoming so people understand what they're getting into. And I think you've done a fantastic job. So thank you so much. Anybody have any questions? All right. Oh, Uwe.
Well, it's not a question, but it's a comment. So we will be hearing about the so-called N of 1 trial later on. Yeah, this is not a compassionate use. It's maybe expanded access or the right to try. Just wanted to tell you that regulators are usually very suspicious about that, so they don't want it to be circumventing or being the fast track for approval. So I think to do that is, of course, it has a different risk-benefit ratio. Of course, if we see encouraging data coming out of that, that is wonderful news, but it will not per se lead to approval as a drug. Regulators do not want these data to be used in a formal drug approval process. It is tremendous supporting information. It's a tremendous encouragement also for us, of course, but it is a different pathway.
Yeah. I'm going to thank you very much. Of course. Appreciate it. Thank you. We're going to cue a video then.
are with Diana and Mike Cooley, and their child Connor has been treated with the ASO for the TUBB4A gene mutation, the first child treated. And Mike, tell us a little bit about how you discovered the treatment.
Well, shortly after Connor was diagnosed in 2021, we were looking for treatment options, and I happened across an advertisement for n-Lorem in the Global Genes Annual Report. And it was an advertisement looking for rare disease candidates, and we emailed them, and they emailed us back, and that started the application process, and that started the process out.
This was very exciting to have such an opportunity and hope. So yeah, it's better than the alternative.
Well, they accepted us for the initial application, but then we had to find a doctor that would be willing to administer it. And then with your help and the TUBB4A Foundation, HABC Foundation, we were able to find Dr. Eckler in Boston with Massachusetts General Hospital, and he agreed to administer the treatment. But that started a process of about 2 years where we would go up there every 6 months to do initial assessments, and then he had to write the FDA approval request, and it took about 2 years before the actual first dose was administered.
3 years.
3 years.
We were scared, but also we were ready for it because, again, yeah, that's the best thing we can do for our kid is make sure we can offer him some sort of hope. And it was a little bit stressful, but it all worked out very well. There was no side effects. He spent the night in the hospital, and everything was great. And he's had 4 doses now, and it's continuing to not have any side effects. So we're all very happy.
So the first dose was September 24, and then he had a dose in October, and then again in December and March, and we'll have the next one in June. First doses are small, so we didn't see anything right away. But since the last couple of doses, we're starting to notice some improvement in his control and his coordination, shaking less, better control of his head. And it's not so apparent. It wasn't so apparent for us because we were, see him all every day. But when we look at old videos from him from just a year, 2 years ago, it's noticeably less shaking and such. And then his physical therapist has noticed he's able to do a few new things that he wasn't able to do before.
I mean, there's different markers that have to be achieved for it to continue, but at the end of the day, the most important thing is to stop the progression of the disease, and any improvements really are a plus.
Well, guys, thank you so much for paving the way for the rest of us, and fingers crossed that this gets to a larger group of people,
yeah.
A larger population.
Good luck, everybody.
So the first ever child treated for the mutation, which is historic. And so I asked Dr. Mandy Nagy to join us from Mass General, who was part of that trial overseen by Dr. Eichler, and she's going to present for us. So thank you.
All right, everyone, thanks for having me here today. Like they said, I'm Dr. Nagy, Mandy Nagy from Mass General. I'm a pediatric neurologist. I work in the leukodystrophy clinic under Dr. Eichler, and I think I have slides. Okay. I've proven earlier in this conference to be technologically inept, so okay, great, thank you. All right, so I'm going to be talking a little bit about what the process of this N of 1 trial looks like and our engagement with the n-Lorem Foundation, which is the foundation that creates the drug that's used in this trial. So my main disclosure for this talk is that I am an investigator on actually 2 trials with n-Lorem. Oops. And that I will also be discussing the use of this ASO, which has not been approved by the FDA for any indication. Okay. So I'm actually going to go forward through these slides because I think we've covered HABC pretty thoroughly at this point. And ASOs, this has been discussed before as well. So let's get into the difference between clinical trials as a group, like Dr. Waldman just discussed, and what an N of 1 trial looks like. So when we think of clinical trials, we think of all these different phases. So there's the preclinical work, there's the phase 1 where you're testing participants, you're determining safety, you move through phases, you include more participants, you start looking at efficacy. It's reviewed, at least in the United States, by the FDA. Maybe you get approval, and then you have post-marketing phase 4 trials to understand what this looks like in a larger group. And so this is a great model for something like a blood pressure medicine, as Dr. Waldman was talking about, where you can get thousands of participants. But in rare diseases, these things look different, and you're obviously not getting these numbers. And in nano-rare disorders, so things like TUBB4A, where you only have a few participants who might be able to get a drug, we start thinking about how can we get access to people faster? And this is where this N of 1 idea comes in, okay? So the NLORM Foundation that we partner with is a foundation that's committed to creating drugs for individuals with severe, progressive, and urgent conditions. So meaning that you're at risk of having decline, you're at risk of severe complications if it's untreated. And importantly, these have to be nano-rare conditions, meaning between, say, 1 in 30 individuals worldwide that they think would potentially be able to use this medication, okay? So there are advantages and disadvantages to both of these, because many of you might be sitting there thinking, well, why didn't we all do this? Why are we not all getting N of 1 drugs? And certainly there are advantages. So it's an individualized drug. It's made for this particular participant. It's an individualized trial and protocol. So some of the issues that people who were discussing earlier of finding outcome measures that might apply to everyone in a trial, it's not such an issue because we have one participant. We selected our outcome measures based on that participant. And so that's how that trial is designed. It is, in a sense, faster. So I think you just heard the family talk about 3 years from diagnosis to treatment. And there was a whole drug development stage in there, which is faster than a clinical trial typically comes. But I think faster in the sense that that's the whole process, but we're coming in the middle of the clinical trial process as well. But the really big advantage is that this improves access for ultra-rare disorders where there is not a trial currently available. Now, there are disadvantages. So there's not a path for an N of 1 drug to be commercialized, to be approved by the FDA, and then used widely. The data we're collecting are helpful, and it's hopeful for the community. But as it was previously stated, this is not something that we can use as evidence for approval of a drug. There's potential bias. So we're looking at one patient. And the way one patient responds may not be the way all patients respond. And it's also resource intensive. So not that clinical trials aren't resource intensive, but there are just a lot of things to think about when you're designing a trial around one person. The other disadvantage, in many cases, is that this is not something that is funded in the same way of a clinical trial. So clinical trials, you have a sponsor, who's the pharmaceutical company, who's paying for most of the study-related expenses. And in an N of 1 trial, that may not be the case. And so there may be some complications around how to get things paid for, how to have the resources to make this trial run. So clinical trials. Many advantages to clinical trials. They allow for rigorous data analysis and collection. There is a pathway to approval. The data is more generalizable, and it offers broader access to the community. Of course, the disadvantages, as we've already discussed, not everyone is eligible. There are inclusion and exclusion criteria. The outcomes have to be based on the needs of the group and not tailored to each individual participant. But again, these are also resource intensive. I put that on both sides. That's one of the downsides of this research in general. Okay, so what does the NLORM process itself look like? I think the process that was described by the Ghoulies is a little bit different than what the process looks like now. Initially, the families themselves were able to reach out to the NLORM Foundation directly. Now applications need to come from a research physician. So families establish care with a research physician who has the capacity to run trials, the capacity to work with a foundation like NLORM, undergo an evaluation, and then that physician actually submits the application. In order to be considered, you need comprehensive clinical information that's submitted to this company. So that includes the diagnosis, it includes the mutation, the symptoms that you have, what organs are affected, what your symptoms have looked like over time and how they've changed. And you also want to have some goals of the treatment. So this question that we've been asking, right? What do we want to improve? How do we want to improve it? What are we actually addressing? And we want to understand all of that before we can actually even submit an application. So once an application is submitted, it goes to the NLORM Foundation, and they meet, I think, monthly to review applications. And they get together as a group and they look at the mutation and decide, is this addressable by an ASO approach? They look at the clinical information provided to say, does this person seem like they might benefit from an ASO? And they look also at the researcher and the institution to say, does it seem like this institution would possibly be able to implement such a trial? Okay, so that's the first step. If they decide, yes, this is reasonable, they accept the application. And then we get into this pathway. Yeah, this pathway here. So the idea behind this pathway is that you're essentially doing the preclinical work and the natural history data or the baseline data collection simultaneously. So on one hand, you have NLORM, which is gathering genetic information, screening for ASOs, so the drugs that they could use, eventually selecting a lead candidate, hopefully, undergoing all of those things that were talked about earlier, like toxicology studies, manufacturing, all of that behind the scenes stuff, before submitting this IND or drug application. At the same time, on the clinical side, we're seeing the patient in clinic. We're collecting data. We're understanding what symptoms are there, how are they changing over time, how fast are they changing, or are they changing at all? And we're treating things clinically as they come up, just kind of typical clinical care. The IND is this big document that talks about both the ASO that's being used, or the drug that's being used, but also the trial and what we're going to do and what we're looking at clinically. Eventually that gets submitted. We have to wait for the FDA to say, okay, you're good to move forward. Then we have to submit it to our own institution, IRB board, that says, hopefully, okay, you're good to go ahead. And then after all of that, you finally get to this point of patient treatment. And so this is essentially the same process, right, that's going on in a clinical trial. It's just for one participant. So in this case, we have an open label, single center, single patient study in collaboration with the NLORM Foundation. Open label meaning they are, of course, getting the drug. So the treatment is with an ASO that's administered intrathecally. And in this case, that means lumbar punctures that are happening every 3 months. This is a dose escalation protocol. followed by maintenance doses every 3 months. And so what that means is, is baseline, we start with a very low dose. A month later, a higher dose. 2 months after that, the maintenance dose. And then once we've done that ramping up phase, the maintenance dose is given every 3 months going forward. So we're looking at a number of a number of endpoints. Our primary endpoints are looking at changes in motor function that are measured on a number of scales that are listed here. Also changes in spasticity and a quality of life scale called the PEDS-QL. We're also, as secondary endpoints, looking at changes in feeding and swallow, because this was a big concern, not just for this family, but broadly. And then, of course, also safety. So, as you know from the video, our patient is I have 5, he's 6 now, a 6-year-old boy who received his first dose in September of 2024. He'd had 4 doses administered as of March, with a 5th dose in June that I don't have in my data yet. Currently in the maintenance phase. This drug has by and large been very well tolerated. We've had a single adverse event, and this is kind of what Dr. Waldman was talking about in some ways when we're saying, you know, what are adverse events in trials? So the adverse event was an upper respiratory infection, so a cold essentially, that is completely unrelated to the drug, but we have to report anything that happens to you when you're in a trial. But otherwise has been very well tolerated. And anecdotally, the therapy teams felt like there was improvement in tone. The parents felt like he was more mobile. He was army crawling around the house more, moving more, and improvements in shakiness, as they noted in the video. So a lot of this is very early. So this is 6 months of data. This is looking at the GMFM, which is a gross motor function scale with a high score being better. And overall, not dramatic changes, but we see some improvements in the total gross motor score, and specifically some improvements in sitting and rolling. We see most dramatically, correlating with that change from the OT, improvements in tone over the course of the 6 months, and also potentially some improvements in language. Notably, this family impact module from the PEDS-QL shows that the family thinks that he's doing better, which is a huge thing as well, and something that regulatory bodies really want to know. So we're still very early on in this course, but I think we've felt that this is really promising so far. We've been encouraged by the response and by the lack of adverse events. But overall, I want to give you the sense that there are advantages and disadvantages to this individualized approach, and many, many advantages also to typical clinical trials. All right, thank you, and feel free to reach out if you have any questions, or I will take questions now.
For families who might be interested in pursuing this, what can you tell the patient community for those that are trying or are interested in pursuing a trial like this?
Yes, it's an excellent question. So the first step, if you're interested in pursuing something like this, is to find a research physician, an institution that is familiar with kids with leukodystrophies, that has experience in running clinical trials, and would be comfortable working with a foundation. And I would guess, if you're not sure of that, you could probably talk to the HABC Foundation, or you can chat with us. And and get an assessment, let that physician know that you're interested, and ask if they'd be willing to submit an application on your behalf. And it's going to depend on a lot of things from your particular situation, but that would be the first step.
In the cost perspective for the families, what is their obligation and what are they required to pay for?
Yeah, that's a great question as well. So the NLORM Foundation is committed to providing the drug free for the course of the trial, but also going forward for life if it seems to be beneficial. That being said, the drug itself is not the only cost that's associated with one of these trials. One of the difficulties of working on this particular type of research is that there is not a sponsor in a classical sense. So in most industry-sponsored trials, you will have someone who is covering all of the necessary procedures, who is covering all the visits, the labs, the MRIs, the anesthesia, and potentially also travel and accommodations like we were talking about. In this case, that's not something that we have the capacity for. So we, in general, have had to work closely with insurance to get things covered where we could, things like MRIs covered under routine care. Where there is not coverage, there is a small financial, essentially grant from NLORM that covers some of the expenses. But it is a question long term of how will all of these expenses be covered. So I don't have the perfect answer. It will probably depend on the particular situation, insurance, and institution. But it is something to be aware of, that there may be costs involved with this that are not involved with a clinical trial.
And then my other question is, how long do these trials last? At what point is it considered viable and works? And then what happens in terms of the insurance company paying for the drug?
Yeah, so the trial is designed for 2 years. So we'll be collecting this data fairly systemically over the course of those 2 years, as defined by the protocol. After that point, it's up to the investigator's judgment, whether this works, whether it doesn't work. And if we deem that it works and the family is in agreement and wants to continue, the NLORM Foundation will continue providing the drug. So the drug itself, the cost of the drug is not the main concern. The concern is, again, how to continue covering all of those other things. It should be covered, much of it, by insurance, but not everything ends up being covered by insurance.
Has Mass General ever gotten to the finish line on something like this? And if you haven't, how do you foresee it playing out?
Yeah, we have not. So we currently have 2 programs with NLORM that are actively dosing, and this one is the one that's further along. So what I anticipate is that many of the costs will end up being covered by insurance as they are being now. Some of the costs around things like anesthesia and actual administration are the ones that I'm less sure about, and I don't have the answer yet. We're still trying to figure that out.
And from my understanding, every hospital or medical institution does this differently. Do you have any perspective on what other hospitals and how they're doing this?
Yeah, we had conversations with a few other institutions and as well as with the foundation and NLORM Foundation directly. It seemed like many were having expenses covered by insurance. That said, there have also been patients or organizations who have done fundraisers to raise funds to cover some of these things to manage costs long term.
All right, we are a little bit behind time, so we're going to take a brief break for like 10 minutes, and then we can come back and pick up with our UnRavel drug repurposing study.
Yeah, so I'm going to get started. name is Frederic Vigneault. I'm the CSO and co-founder of UnRavel Biosciences. UnRavel is based in Boston. We're a spin-out from the Wyss Institute at Harvard University. And originally I'm from Quebec, so it has a French name. The agenda for today, so I have a disclosure, and I'm gonna go quickly over the UnRavel. I had like some section of this, but in the interest of time, I'm gonna like fly over this and go straight to Redshift, which is the project we're doing with the foundation here. Disclosure quickly, the first one is just, we're a for-profit company. We're still private. I own a bunch of shares. Fight HABC did support financially the project. Doesn't mean, they endorse the company. And, important point, I will be discussing drug repurposing. I have a couple slides on using vorinostat on Rett syndrome. We're not going to be discussing off-label use for your group. And the reason is this is still considered exploratory project, right? It's a research project basically which could help other group, let's say with the mouse studies or the IPS, decide which existing drug, for example, to test in a model before you will do that in a human being. Also, we developed a tool which is using AI basically to do drug predictions. So, whoever participated in the study, and I'll get to that, are the people we use to make the prediction. So it might not work for everybody, until you sort of test it. So that's the part I'm gonna go over quickly, but basically the idea with rare neurological disorders or the drug development in general, is most companies start with the target. And when you have gene therapy, that's fine. You know who your target is, and that's what people have been doing. But most of the time for complex disorder, and we started with Rett syndrome, which regulates 1,000 genes in your body. Then we still don't know. People have been working on it for 25 years. They still don't know what it does or how it works. So this is basically bound to fail. And this is 95%. This is the success. This is the failure rate for all drugs in the pharma industry. So we set up to think differently. Let's start with the patient instead of talking with target, right? So we start with the patient, then we ask, we want to answer a clinical function first. Then we're going to figure out how does this work to make a new drug. So that's how we started UnRavel, right? It's like using technology to answer a biological riddle. By the end of the day, we're a pharma company. And patients, as a result of you have a mutation. You've been growing with that mutation since we're like an embryo. Even if you're like a healthy person, you must have variants. You're all different. Some people are taking medications, that's going to affect you. So 2 years old is not the same as a 20 years old. So you can't use genetics really for just your gene sequencing for that. So what we do is use RNA, because RNA is dynamic. It changes with you. It changes with your sleep pattern, medication you're using. And that's what we decided to use for doing prediction. And the way we do it, so you probably have heard other groups like at TUBB4A, by example, doing drug repurposing. They also use RNA, right? They're going to look at a gene going up, and they're going to try to find a drug that's going to make the gene going down. The example of that is if you have a roadblock in your system, and you see a gene going, this is a gene going up, then you're trying to get your gene going up is called a reverse signature strategy. You're basically going to try to create a detour around, and then people might get off the map, basically. So that's why the reversal of those gene signature never works or rarely does. So we use a different approach. We're basically closer to what Google Map does. So your traffic jam here, we don't really we're not trying to remove it like gene therapy will do, or we're not trying to detour it. We're just trying to find the best path to get, to your destination. So what we do is we compare a patient, RNA network, which we call a disease state, and we try to compare it to a control, which we call the healthy states, and evaluate all the different paths you can take, right? And then find the best one. We basically do like Google Map is doing. We evaluate like 40,000 different paths. We rank them by the best one to the least likely to work. And that's the one you go after, basically. And in our case, it's like to make a new drug. So I mean, this is a heavy slide, so I'm just going to skip it. But basically, we build like a large network. We have like millions of interactions that we're looking at, so not just a few thousand genes. In a house, because we want to look at function first, for Rett syndrome, for us, it was important to look at seizures, motor functions, like sort of behavior. These guys have been excellent at picking seizure, autism symptoms too. They have schooling behavior, so we know when they don't do schooling behavior. Sleep. And the idea is that you can use existing molecule basically to go do a study in a patient very quickly. You don't have to develop something new. You already have a safety profiles. So for us, it's de-risking a new target. Basically what I just say here is like an existing drug. De-risk, validate your program. Most pharma industry doesn't really do what we call first-in-class, which is when you make a new drug that's never been tested like for a gene or a protein before, because the failure rate of that is even higher than 95%. So Rett syndrome is what we started with. It mostly describes neurological disorders, but affect like the whole body. And we did this thing where we use RNA-seq data, plug it to BioNav, tested it in the tadpole model, then mouse model, see that it was working. And this is using like Vernostat, an existing molecule. There's a boy with Rett syndrome currently taking the medication. He's been on it for more than 16 months, I'd like closer to 18. And in the process of doing that, with BioNav and sequencing the mouse again, we did find how Vernostat worked for Rett syndrome. And it doesn't work the way Vernostat is known to work. We found like a new mechanism, so we've been building like a new molecule basically that's specific for that. It's safer and it's going to be better. But this is like your new drug development. This is the thing that's going to cost like 15, 20 million to get to like an IND basically. While this one exists, and we manufacturing it for like enough for like $500,000 for a clinical trial. And I'm just going to skip over this. So I'll get to RareShift. So RareShift is how we call our program with foundations. When first foundation heard that we're using like existing molecule, like we have like 40,000, most have never seen a human, but we have most of the approved drugs. We can add anything. Doesn't have to be a small molecule. We can add ASOs. As long as we have RNA sequencing, we can add it to the simulations. But then they wanted like to focus on can we predict a repurposable compound or nutraceutical or supplement or something like that. And that basically feeds into the platform I just described for UnRavel. So the way we work with Foundation, the way we work with this one is we collect samples from the patient and we pick nasal swabs. And that's going to seem strange, and especially for leukodystrophy. And leukodystrophy, the HEBC is like the first one we're doing. So we're curious to know how this is going to work. But we've done this with serial individual and foundations, where we collect sample from the patient and the control. We do the BioNav prediction, and we deliver a report with all of those things. We share all the data. We meet with the foundations, the family, answer any questions, and plan for the next step, right? So nasal swab, that sounds curious. It's tiny amount of tissue. It's different type of tissues in there. We do pick all the genes, coding, non-coding. And there's a Synapse gene ontology database, list all the genes that are supposed to be mostly synaptic. or just in the brain, and we see all of them from the nasal swab. We do see tubulin beta 4A is in there. It's a very low level dose, so you'd expect it to be higher in the brain, which means that we're probably not picking like oligodendrocyte, by example. So yeah, that brings to the next question. Will this work with your gene, right? And so UnRavel and the BioNav doesn't try to upregulate or downregulate that gene in particular. What it's trying to do is like find alternative path, by example, or a mechanism that can be compensatory. And it's going back to, you've been growing with that gene, with that mutation for some time. It has its effect. And we know like the model dies pretty early, and yet your child is still alive, right? So there's enough plasticity in there that we can play around it. Again, leukodystrophy is new to us, so maybe the best case scenario is going to be symptomatic. For Rett syndrome, we saw that vernostat is not a cure, but in the mouse model, it was better than gene therapy. With the bio, we know it's not a cure. Like we can see the bio improving, but and the idea is, then we get more data and we can do something else with it. Yeah. So this is another slide on the sort of BioNav, but basically you have those 40,000 molecules. Most of them, we're going to predict them all. We use them all to do simulation. We rank them from best to worst. Most don't get predicted because they don't affect your network. It's all specific to each of you. And the way the study works is some of you received nasal swabs, right? And we ask a patient and a same-sex relative to get swabbed, and we sequence that. And we did it with, in this case, with 18 patients and controls to get sort of like an idea of what the population looks like. So we had to upload the slide like 2 weeks ago, so we didn't have the HABC data yet. We got it just like a few days ago. I did upload the slide like yesterday. So I'm going to go over this one quickly, but the idea is like, this is a correlation plot for another disease. We're used to work with complex profile, complex population. They have different mutations, sex, age. And what you're going to be wanting to look at is like clusters, things where like the color seems to be matching better. So I'll just skip over to the HABC slide. Oh, this is the old. This is the uploaded slide deck. This is not the uploaded version, the updated version. Yeah. Ooh. Sorry. But yeah, so I'll explain a bit what the correlation does. It's a statistics, right? So like I say, we sort, and it's easier to show, I just don't want to show any drug, but basically we sort a list of drugs, like names basically, and they have a specific order. And that's for each of the samples. So each patient we sequence has its own list, right? And the correlation is, another analogy is if I ask you to make a grocery shopping list and ask you to make a grocery shopping list, what are you going to put first on your list and like how many items, and then you're going to have your list, right? I'm curious, what's the first item on your list? Salmon. So you see what beef is salmon here. Same category, meat, but they're already different. And then we could do it, maybe it's gonna have 20 items, maybe it's gonna have like 10 items, and then you compare. And the correlation statistic is very strict for that. We have like other tools we can use, better statistic to compare different variable. But the idea is you compare the list in the same name, same rank, right? So it's very hard to have 100%. Will be very surprising. So the color code is here, like correlation. So it goes from, you have 0, which is like no correlation, and 1 would be like 100% correlation. And -1 would be 100% anti-correlated. So anything that's like in the like 25 to 50, we're very happy with, especially when you have like 100, like your list, grocery shopping list of drugs is like 100 different names, right? To keep going with the so basically that's what you look at. And we did if the color is a bit hard to see, you look at the sort of the cladogram here. We do have like 2 groups. There's one that's stronger at the bottom here, and there's one that's a bit more diffuse here, but we do have 2 groups, which, you know, for 18 patient, a disease that like a variety of presentation and mutations, it's pretty good, it's pretty encouraging. So for us as a company, once develop new drug, it means like we probably have to think about 2 programs. But we have the all versus all, which is basically lumping everybody together, see how they're doing against each other. This seems to do well with the first cluster, but basically there's something in there that should work for everybody. But it's possible, like I'll list like the everything bagel, and that group might like the everything bagel, and this one might like cinnamon bagel better, but you know, it's still bagel. And then we have like some other information we can look at. So here we only, I mean, everybody has the same, not the same mutation, but the same gene mutated, but we could refine it, right, by, let's say, who has a 249N mutation versus who doesn't, or who has a mutation like we saw with the natural history study that does like a late infant versus early. So we could do that too. So didn't do it here, but we can. Because all of you who participated, you did provide the genotype information, so we can do that. The second one is the sex. We had 6 girls in the study, like roughly 33%, right? Doesn't seem correlated by sex. Age, there's a small bias. That group seems to have more younger people. So, you know, maybe that makes sense. The average, so we had you can see in the list, the youngest had 2 years old. The oldest is 20, actually. So average 10 for that study. But yeah, that's the only slide I'm going to show for HABC. The report is still being generated. It takes us about a week or 2. I mean, getting the swab takes about a month, sequencing 4 to 6 weeks, and then this you know, a week or 2. We try to automate most of it, but there's still like some manual input in there. But yeah, in the report you get everything. But the foundation is going to look like Michele is going to get it first and meet with us, involve anybody else. Yeah, that's basically like a list of everything we provide. So I'm not going to go through all of this, but we share everything with you, like RNA-seq data, like if CHOPS wants to get the gene expression, we give the gene expression. Everybody's identified. We're not going to re-identify anybody unless everybody accepts, like all 18% accepts to be identified. Yeah, so one thing we not do is tell you what to do, basically, or what to take. You know, that's your clinician that can do that. Like we cannot treat patient. The foundation cannot treat patient. Only clinician can do that. So we've done that a bunch of time. We've sequenced over 1,000 patients, 1,000 control all over the world. We're using swabs that are shelf stable. 60 different disorder, but like I say, we're new to leukodystrophy, very curious to see how that's going to work. Going to skip a bit over that. This is an example we did recently. This is a mitochondrial disorder. So we have a couple of videos. The family actually want us to share this. They're trying to promote their effort. But this was before the prediction, and this is after. So on the first one, like you could see, like she wasn't able to sit no tonus. Here they're making sure like she does hold, and this is like only 3 weeks after. And you don't hear the sound, but basically the therapist is recording this and saying like he cannot believe it. It's not even a drug. Those are just supplements that she took. Yeah. Yeah, so just to close, basically what I mentioned is, we start with patient, we did the swabbing, we did the prediction. Ideally, the next step is like test this in a model. Normally, that's what we like to do, and work with clinician and your foundation with the next step. I mean, you guys have great models for this, so it will be interesting to look into that. We can, I mentioned swab, but the Hunter model was based on was built by changing gene expression in human cells and testing drugs to see what they do at their level in human cells. We like to sequence mouse, especially like brain samples, to compare how they do with the patients. Same thing with the iPS cell. Really, nothing was built based on swab. It's just a way for us to get like the disease state. But the disease state can come from any model, and ideally we'd have both. And that's the team. Like I say, we sequence a bunch of people. We have 60 projects. We're 10 people. So we've made this very efficient. We have half the team is computational, half the team is wet lab. So that's that. Yeah, thank you.
Thank you, Fred. One of the things that I want to make sure everybody understands is once we get the results and the full study back, our next step is to bring in clinicians who are familiar with TUBB4A, and we will be presenting to them the supplements and or the drugs that come out of this to see the viability and how it can help and any further analysis that needs to be done. So we're very conscious and mindful of that, the need to bring in the clinicians to see how it impacts the condition and the safety and all the other factors. And from there, we'll provide some details, and then the individual families would make their own decisions whether they want to use it or not. Thank you. Anybody have any questions? Yeah.
So once you guys get that data back from the clinicians, would that data be publicized for everyone in the community or just the participants of the study?
No, once it's formalized and we go through a process of formalizing that, and again, I don't know exactly what that looks like, and maybe Fred has seen it, but it would be available for everybody to have access to. But again, I had emphasized you have to work with your own physician because they know your child best, and we would not be making any recommendations other than this is what the findings are, and you can take it from there.
Yeah, we let the foundation decide what they want to do with the research, the data. We like to know who's going to get it, but we're not going to prevent you from like sharing everything. You can actually find, we don't share a report like on our website, but you can find like posters, publication, videos. Sometimes we have families who want to like highlight that it worked for them. We have videos of that too. They want some groups like they want to raise awareness, so like they're glad to have like a YouTube video sitting on their website that other people click, right? So yeah, you can, like we have their UnRavel.bio website. We also have like a Rareshift forget if it's bio.com, but was on the slide. But both are accessible from each other's. So you see everything. You see the whole Rareshift process, how much we charge for that, these type of things. Yeah.
So have you guys gone all the way through from beginning to end? Where are you with the Rett syndrome? And
so Rett syndrome, we're hoping to start a clinical trial this summer with 15 patients. It's going to be done in Colombia, the country. We're going to use an N of 1 approach where every patient is going to be its own control, which is not something the FDA likes to do. We did meet with FDA. We have like a pre-IND meeting, so we know what they expect. But we're using that sort of like a validation, awareness, that getting observation. And that's, again, going back to using existing molecule. You can do that too, right? You don't have to start a clinical trial. Like a clinician, like a child, by example, could decide to do an observational study under IRB and collect some data before deciding to spend more time. Vernestat is a different story. There's a commercial opportunity for us just because the drug costs, manufacturing process, and formulations is not compatible with Rett syndrome patient at all. So we had an opportunity to make like a better product.
But there could be instances where a compound could be used immediately
Yes.
if, again, the physician following the child agrees with it. But in your case, you had to go through a clinical trial because more analysis and research needed to be done. Can you explain the differential?
I mean, we have the second project is X-linked dystonia Parkinsonism that's endemic to the Philippines, working with that with MGH. And the drug we found is available in every shape and form. It's cheap, like we're never gonna do anything with it. So MGH wants to run the trial basically. So, you know, we're just assisting. There's another one which is closer to what I just mentioned for pediatric dystonia, and started with one patient where a clinician did try one of the prediction. The boy did really well, and now he has 5 patients with the same mutation. So he wants to do sort of like an observational study using combined brain platform for, you know, to record data and so on. So the foundation basically, they're pretty new. They're just like a month old. So they're looking like for surveys or like tools that they can use to make that case. However, that drug again exists in every shape and form. It's cheap. So for us, there's no like commercial opportunity. So we're just like helping. Yeah.
And what are the downsides that families would need to consider taking this path if there was something that came out of this?
It's a good question. You know, it's like even though it's an existing drug, there's like a known safety profile. You still have to be, I guess you still have to be careful, right? It's like with everything else. It's possible that the insurance are not going to, it's likely the insurance are not going to cover it too. So, you know, if the drug is expensive, then, you know, it's possible not every parents are going to be able to participate. For us, you know, we're giving you data, right? We're like RNA data, drug prediction. So, and what we like to do is like some follow-up, just so we can like sequence again, maybe at our own cost, and figure out, like, validate the targets, see how the drug actually worked in this patient. So, I mean, I'd say like the downsides are minimal. One of the biggest limitation is when you have, is like, we do meet sometimes with clinicians, don't want to prescribe anything that hasn't been approved for that condition, right? So that's a bummer for a lot of the families. We try to help them meet with other clinicians that are a bit more willing. But you see, we also have like families that just don't want to try a drug, right? They're gonna fix on supplements, and we might not have one, right, in your list. There's always, so we have yet to have that, but there's always the risk that we're not gonna find anything that's easy to use, right? There's always that. So yeah. I mean, I feel like it's a risk because like you're paying for that, right? So you're taking a chance that you're gonna find something. We try to make as cheap as possible. Like, we make no profit out of you. It's just paying our cost. We're like a tiny company that's started like at the wrong time in the economic environment. I mean, we were talking about that over lunch. Yeah, it's not easy. Yeah, it would be nice, you know, when the big pharma company pays all your travel costs for a study, but most the young startup can't do that, right? So yeah.
Anybody have anything else? All right. Thank you, Fred, very much. Appreciate it. All right, Kim, you're up.
Hi, everyone. My name is Kim Hindery, Executive Director of the Child Neurology Foundation, but this isn't professional for me, it's also personal. I have a daughter who has some unique healthcare needs, diagnosed at 1 with polymicrogyria and schizencephaly. That's a mouthful, just like all rare conditions are, but basically means that she has a cleft in her brain in the frontal lobe with small folds coming out of that cleft, where neurons can't make connections. So therefore, mental health is a challenge because that part of your brain controls your mental health, ADHD, anxiety, all of those things. But then just recently, after 4 years of a long genetic testing odyssey, we found out she's one of 2 in the world with a rare leukodystrophy gene as well. So that's why we started to see a little bit of decline in what she's able to do. So since she's one of 2 in the world, we really have no idea what's going to happen, right? It could be a spectrum. She could decline rapidly. We just don't know. But I think ultimately what has really kind of pulled our family through the last few months is a community of support. Our community is not yet formed. Obviously, there's 2 of us, but there's less than 30 with the sister gene. Immediately found them on Facebook and found the other mom who lives an hour and a half from me, whose child has that same gene. She's been studied at CHOP, and ironically, the CHOP team has requested to see my daughter as well. And so we will be meeting with Dr. Vanderver on Thursday. So strange, because, you know, it's taken everything I had to be professional and not go up and go, like, I went in early into some of my questions, you know, like, wait till Thursday, Kim, you can do it. But it is a fascinating field, and being connected with other families truly, truly impacts our mental health. It really, you connect with people who get it. You learn things that you never thought you'd learn. And so our family support program at the Child Neurology Foundation really aims to get families connected. You will have a very small community, but the challenges you face are not rare across the community. There are lots of other rare conditions that experience some of the same challenges you do. And so while you feel like you're small, you're part of a much larger community dealing with many of the same issues. So our goal really at the foundation is to be this center of collaboration and convening for patient advocacy groups, clinicians, families, researchers, so that we can make the biggest impact possible. I have nothing to disclose. Today I'm going to go over our CNF's mission, why we do what we do, some of our programs that we have as far as resource navigation, family support, education and resources, and how you can connect with us. You are already part of our network, and so I think somewhat recently have joined our family support program. We right now have over 95, almost 100 now, advocacy organizations that have come together to provide social service supports for families, and there are benefits that we offer at the foundation just for being a part of our network. It's free for any patient advocacy group because we believe that supporting patients should be free. There should be no barriers in that regard. So again, this is just our mission. I'm going to go through that. So we really believe, again, no one should be facing this journey alone. We have a large community. We continually say, oh, my daughter has a rare condition, right? But the rare disease community as a whole is large. It is large, and we can really cross-collaborate to make sure that patients are served, their extended family, their family is served, and so that's exactly what we do. We try to bring people together so they don't go at it alone. I just went through that. So here are just some of the resource navigation programs that we have, or things that we can do for you. We do have a social worker on staff. She speaks English and Spanish. Vivian is her name. She is wonderful. She is an expert at finding things in your community around the nation, from mental health services to financial assistance. If you need help getting to a clinical trial, if you need help with that transportation, we can help you. We have partners to do that, that even give vouchers from, the airport to the hospital. So that should never, ever be a barrier to participation if that's something that you are interested in. We can even help with employment assistance or financial assistance. Just don't ask us to pay your mortgage the day before it's due. That's really hard to find, okay? I will say we have done that before, but it's difficult. It's really hard. So give us a little bit of lead time. We can also refer, if you are a clinician and you have a family that comes to you, you can fill out our assistance request form on our page, and we can get the families to the advocacy organization that most directly aligns. But then we will take care of, our social worker will take care of any of the other social service needs. But it is really important that we get you connected, obviously, to your advocacy organization so you can connect with other families. We have peer support as well. I'm not sure if any of you have met Brad Thompson. He is an absolute joy. He is a licensed counselor as well as a dad to a daughter with complex healthcare needs who's in her 30s. So he's lived this journey. I call him the salt of the earth, and he has a fan club, literally, and he gets really embarrassed. But he does a great job of putting this life into perspective. We deal with challenges that no other families deal with, right? Like, I shouldn't say no other. There are a lot of other families who deal with these challenges, but a typical family doesn't have to deal with some of the things we do. It's a different kind of life that we live, and he really helps you cope with those challenges. Here's kind of how our family support cycle works. So a family needs help, they reach out to us, our social worker contacts them, picks up the phone and calls them, unless you say that you prefer email or text or something else. Once you contact us, we find out your story. It's completely confidential. We don't share your story with anyone. A lot of times people will be referred to us for one thing and they need a host of things, right? Because that's just how it is living this life, right? We constantly are in need of a lot of different things. We'll get you connected to another parent, the patient advocacy group. Obviously, you all are connected to a patient advocacy group. We can help find educational resources if you need help finding a specific doctor. And more importantly, the other thing that we do is provide education. I think, you can never have too much, whether it's from learning about the IEP process to how do I transition my child from pediatric to adult care. We have resources on all of those types of things. We have a family support handout here with a QR code. You can go to our website as well to get that, or I think you all have one on your website as well. You can directly get connected to your foundation. Oopsie doodles. But here's some of the education as well that we do have on our website. We are doing extensive outreach to schools because a lot of times what ends up happening is that students have educational diagnoses of autism or CP. No one ever refers them for genetic testing. We don't know these kids exist. They can start to decline and people go, oh, that's just, part of autism, or that's just part, and then it delays treatment. Then it delays therapies. And so we need to identify them sooner. We went the route of contacting clinicians and letting them know, hey, we have a family support program. But then we realized clinicians don't see all these people. But who does? Schools. Schools see them every single day, sometimes more than their own families see them. And so we really want to make sure that we are getting in these schools, letting families know we exist. Genetic testing exists. So if you have a school where your children belong, let them know about us, because a lot of these families are walking around, oh, my kid just has a learning disability. Is that really it? Maybe not. So there's a whole population of people out there that don't have the information that they need. So these are just some of the educational topics. I won't go through them just like that. But some of the resources we have as well, a child neurologist new visit toolkit. If you have a neurologist by you and let's say they're just not meeting your needs, they don't understand your condition, you want to go to a new neurologist, or if it's the first time, if you guys get another family that enrolls in your organization and they're looking for a neurologist, we have a new child neurologist new visit toolkit. I actually have used this. My daughter transitioned from Nationwide Children's Hospital to Cincinnati Children's Hospital, and the amount of information and how long it took me to gather that is unreal. And this really helped me do that. So great form. We have a respite care notebook. So if you do have someone coming in for respite care to your house, you can make sure everyone is on the same page. They know what medications your child takes, when, what soothes them, all of those things. Again, if your child is transitioning from peds to adult care, there's a transition checklist that is actually just integrated into Epic as well. So if I don't know if there's clinicians here, but that is amazing, and will actually generate a summary for the next clinician about your child. And a seizure action plan. Every single school should have this on file for your child. So if you do not have this on file at your school, and every teacher who comes into contact with your child does not have this, they should. Because this is incredibly important to making sure our kids are safe. If you want to get connected, here are some ways to contact us. You're welcome to email me as well. Always happy to support anyone. One of the initiatives that we have coming up this next year is we are hosting 25 courses for families. Our course is actually called From Surviving to Thriving, and it is a course for caregivers. We'll go over topics such as grief. What does that mean? It's not just grief for the loss of a child, anticipatory grief. You know, I thought my child's life was going to be like this, but it's not. I thought my life was going to be like this, and it's not. Relationships. How do you handle the holidays? Does your extended family get it, right? Schools, all of the things, basically, that are challenges in your life. Brad Thompson, as I spoke before, he is the moderator of that course. Fantastic, and you can do it amongst your own community. So if you ever have any questions, please let us know. Please subscribe to our newsletter. That'll give you all of the updated information and programs that we have coming up here in 2026 already. Can't believe it. Someone told me summer's almost over, like for our kids. Like it's halfway already. I don't know where it went. So, does anyone have any questions for me? I went through it kind of quickly.
I wanted to share that CNF is actually working alongside the ULF in providing this exact sort of training and support for their ambassador program, which
Yes.
is maybe you could talk a little bit about that.
Yeah, sure. So we have family support specialist training that is really helpful. If you, and I don't know if you have ambassadors or not, or if you just, do you utilize their ambassadors?
I'm one of them.
Yeah. Oh, okay. So you're the only one for the HABC though. Okay, got it. Oh, awesome. Okay. So ULF has an ambassador program to support families. They can utilize all of the resources here, but the training is for the ambassadors is a 6-week training, and it really goes over how to support families throughout this process, how to lead support groups, how to deal with grief with families, relationships, setting boundaries, developing empathy, all of those skills and traits that are really important for ambassadors to have when they're helping families. And that doesn't mean that these ambassadors are going to be able to answer all your questions immediately, but it does mean that they're going to be there for you to listen and to be able to find answers for you. We understand and we know that it takes a lot of time to find resources, and sometimes you just don't have it. You need to have it, but you don't have time to have it, right? Or to find it. And so the ambassadors are there for that, and having that group of people there to support you is really important. One of the things that I've noticed in our rare community, and I am so guilty of this myself, is asking for help. We're really bad at it. Really bad at it. We say we don't have time, or we're embarrassed, or all of those things, but it is the best thing you can do. When I found this one other mom, I felt like the world opened up for me, right? It's a completely different feeling to have someone by your side walking this journey that you can text and go, hey, how is that treatment going? Or, hey, today really just stinks. Or guess what my school just did for my child? They adapted the recorder for my child, and now they can participate in music class. That actually happened to me. I was like literally crying because no one has ever done anything like that for my daughter. But those are the kinds of people you need beside you. so I just can't say enough about reaching out to the ambassadors and building those relationships, because your community doesn't feel as small anymore. We're all dealing with the challenges. It's not just local to HABC. And ULF makes it larger. And then if you participate in some of our other courses that we have, you'll see it's even larger. There are a lot of people out there and a lot of different organizations that have the same challenges and deal with the same complexities. And some of them have amazing resources. And this next year, we're developing a journey map with the resources of our partners on it, so that you can see along the journey, if your child's young, here are some resources that might be of great benefit to you. If your child is, in the middle school years, here are some great benefits, some resources that might benefit you in that stage of your life, or even, into adulthood as well. At the foundation, it says we are child. We're not just, we're lifespan care. So that's probably the first mistake that people usually make. And I always tell people, my child is gonna be my child when she's 30. She's gonna be my child when she's 40. She's always going to be my child. Neurology is a second thing that people always say, well, my child doesn't have a neurologic condition. I know that's not the case here, but for professionals in the room that don't know our organization, even kidney conditions, if your kidneys start to shut down, you're likely going to have some sort of neurologic side effect, right, or issue. And so a lot of these conditions, I just started saying disability, lifespan disability care, because that's the best way to kind of think about our organization and the people that we support. And we support the whole family. So siblings included. There is a sibling workshop in Cincinnati in a couple weeks. We will be posting it on our Facebook that you can enroll in. So if you have siblings of some of your children, an amazing workshop there. We can't forget about our siblings because they're important. And then we're also looking into next year marriage retreats for families. So that is the other, we have a lot of things going on. Lots of things. So do you have any questions for me? Any other questions?
I can't say enough about how broad this is reaching in terms of how you can support families at whatever stage they're at, whether it's initial diagnosis and starting out, or what doctors in this city, or does anybody know a specific insurance company that, or what insurance companies do pay for this?
Yeah.
I mean, it's huge. So it's instead of us, each one of these rare disease communities recreating the wheel and doing all of this themselves, it seems very logical to me that an organization like yourself, you guys are great resources.
Yeah, I mean, we have clinicians that have referred families to us, and they said, this family has this gene. Do you know of any other families? And I looked it up online because I had never heard of it before, which these days it's sort of rare if something comes across my desk and I haven't at least heard of some of the letters in the genes, right? So I looked it up, and it was associated with craniofacial abnormalities. So I called one of our partners is the Craniofacial Association. So I messaged their ED and I said, hey, do you know anything about this gene? She goes, no, but I'll ask one of our board neurologists. I'm like, awesome. So she did, and the board neurologist says, yes, I know that gene, and I have 2 other patients with it. They didn't even have a foundation yet. No one else, but we can get that family connected. And who knows, maybe there will be a foundation next year, right? But that's how they start. People have to be connected to each other. So just all of that to say, with our network, it's difficult, or it typically doesn't happen that we can't find something for someone. With that being said, we know there are gaps in care for our community. We know there are. One of the things that we do in our family support program is we collect de-identified data on the things that on the back end. And what we do is we look for what services people are requesting and we can find them, and what services people are requesting and we can't find them, and where in the country. So that we can identify those desert areas, we can advocate on behalf of our families for those services that don't exist, and make sure that we're creating programs that meet the needs of our community, because ultimately that's what's important. So all of our network partners, we do collect that data from our entire community, and like I said, we have a lot of advocacy organizations in our network. It is free to join, and then also for any new advocacy organizations, their disorder gets listed in our directory as an entry, and that directory is written by a child neurologist and vetted by our content review committee, which is a board of 5 neurologists, and has literally treatment options. It has everything literally about the condition, including what advocacy organizations to go to that could help support you, and family stories. So we're always looking for family stories too, to let people just become more aware of all the conditions that are out there and how they can support. And you never know who's going to see it, right? Our connection of researchers could see it and go, yep, I want to support that family, or, ooh, that sounds exactly like this other condition that I'm helping with. Even here at this foundation, I met another executive director of another foundation, and she was like, hey, here's how our condition works. And I was like, that's how my daughter's condition works. Oh, that's how this one works too. And so maybe we could get someone that could study these together, because they all have this common trait. So that's how we're gonna make things happen for our community, is by working together and cross-collaborating to find these treatments, to find these things that help. And maybe, just maybe, it's not a cure, maybe it's something like a drug that could change the quality of life of our children. That would mean everything to me for my daughter. I want her to live a more comfortable life. So sometimes it's not always about the cure. Sometimes it's really about quality of life. And those are things, you know, as some of the speakers earlier were talking about, has to make sense and has to have meaning for patients and their families. So critical.
Thank you very much, Kim. Appreciate it. All right, we're going to backpedal because we never did the first part. Because we went right into the research with CHOP. So, all right, 10 years. We're actually celebrating our 10th year. So we started out in 2015, and we're in our 10th year celebrating. What we're still waiting for is a treatment, but I think we've come a long way for such a rare disease. It's a quick summary of what we've done. I did want to also acknowledge that the HABC Foundation UK was supportive of the drug repurposing study, and also continuing to support scientific and families conferences. We do have a RareX patient database, and we have completed an FDA listening session. And then, of course, the patient support database and the ULF ambassador support, which Kim just went over. But I'm going to scoot over to acknowledging the other advocacies that are supporting the condition in order of the Foundation HABC, Foundation UK, Fundación TUBB4A Spain, and then, of course, the Kinslow for TUBB4A Foundation. But lastly, I did want to update everybody on the AAV gene therapy. We have been under a sponsored research agreement with UMass now. We're in the 4th year with them, and it's continuing to be challenging. As you probably know, and I'm sure Amy can speak to this, AAV gene therapy is a one-time treatment, and it's successful or not. So there's a lot of work involved in ensuring that the toxicity and the safety is paramount. So we're continuing to support UMass's work. They could not join us today because they are restricted in what they can do now because of the NIH funding challenges. But at this point, we've extended our agreement with them for another year. The last round of the vectors that they developed proved to be unsafe. Toxicity is the continuing challenge. And so we have retained them, extended the agreement, and they have picking up and redeveloping 2 new vectors. and we're hoping that they show some success in the next 6 months. We're also are working with a private company through our board of directors, have affiliations within the gene therapy space, and they are doing a review of the work to date, and we're hoping to connect the two of them so they can collaborate. Sometimes researchers in academia may not be as, I guess, focused on some of the stuff that maybe the private sector might be, so we're hoping that they can add some insight and maybe help move that forward. So I did want to make sure I touched on that, because I know it's a big permanent cure, one-time treatment, but a lot more involved and a lot more invasive. Any questions? All right, do you want to, yes?
Does anybody want a primer or like a 3-second
Yeah, I'd love it.
why gene therapy is so challenging?
I love it, thank you.
Okay. So first of all, what UMass is doing and others is very exciting with AAV gene therapy. So the idea behind the gene therapy is that you take the TUBB4A gene and you package it in a vehicle or a vector and you deliver it to the body. And if you deliver it through an IV, it may not get into the brain. So then if you have to deliver it to the brain, and let's go back to the IV example, you have to give very, very high doses to even hope to get to the brain, high enough doses that it can impair the liver, and there's a lot of challenges. And so I think that's one extra problem that we have in our neurologic diseases, is how to get that vector into the brain. If you don't deliver it IV, you can deliver it directly into the brain, but that requires surgery. And so there's additional challenges, or you can maybe direct it into the spinal fluid, but is that going to get into the brain tissue? So people use the example of a vehicle or a car and driving it to the right place. The other challenge is that the vehicle, the car, activates the immune system for everyone. This is not specific to HABC. And so then your immune system attacks the new vector and the car. So this is why gene therapy is so challenging. We're talking about in vivo gene therapy. There's also ex vivo gene therapy. That's another thing for another day. But this is why I think it's progress. It's one step forward. It's 3 steps back. And the UMass group is fantastic. And I know that they're really doing everything that they can to try to find something that is safe that can be delivered. So I don't know if that helps, but I just wanted to explain a little bit more about why this is taking so long, is so challenging, and yeah.
Thank you. I mean, it's a long process, and we've been at it now for 4 years. And with COVID, we started at COVID, and so it was a slow start. And but it's a stop and start process. So we're hopeful that something comes out of this. Otherwise, we need to reconsider what we're going to do next. To introduce you to Ari Rosenberg. He's actually a recently diagnosed family member, guardian of his brother, Benny, and I think some of you may have met him or seen him on TikTok, but he's doing some great work in advocating and sharing the word. So I asked him to speak a little bit about communications and AAC.
Is that available online? I can say it will be. Yeah. Okay. So you have a handout about different AAC devices. I'm not an expert. I'm simply an older brother to someone who has HABC. We only found out last year. So for years, it was always undiagnosed leukodystrophy. So when we found out it was HABC, it brought us to you awesome folks, and now we have a community. And we decided to start creating videos about our journey. One of the big things when Ben came into our care was figure out a way to get him to communicate again. Ben could talk up until he was, 13 or so, 14. That slowly went away. Sign language worked for a while. He had some devices that he refused to use at home, so it collected dust. But he had stuff, he has language, but he wasn't saying full sentences to us. You know, it was always like a handful of words, little banter between brothers and stuff. So we knew devices exist, and I'm sure a lot of you know devices exist. So I'm not going to go through every single one here, but we tried eye gaze, and that's something where your eyes can kind of pick out things on a screen, and that's something that's very useful for people who have full control of their eyes and good vision. Ben does not. Also, there are some other ones where it's trigger-based, based on head movements, and those don't work as well. So one of my missions was to create something for him. So with ChatGPT, I created some custom software. It's basically 2 button switches that are on his headrest, because Ben is now nonverbal, completely quadriplegic, aside from using left and right head movements. Shorthand, we would ask him yes and no questions. Right is yes, left is no. But, you know, beyond that, you know, that was all we had. So I had to develop something. And just to go back in time really quickly, when he was at home, that's kind of what he was used to. And as you lose these abilities, you'll notice that, well, Ben and kids get frustrated. You know, they don't want to try anymore. So how do you motivate them to want to try again? Well, Ben's was not some new therapy. It was literally, I want to change TV stations again. Like, that's all he wanted. So we developed this system, right and left, head clicks, and he had basic communication. Yes, no, I want this, I want that. Maybe 64 phrases, but he wouldn't use it all the time. It was like, how do we get him to use this more? So I developed a way for him to select all of his TV shows and movies in one device that can go back and forth between that and his communication. From there, I was like, what else can we do? So I developed a bunch of custom games for him. So he's a little dyslexic. We got him word puzzles. He's spelling now. He's saying things on a predictive keyboard we developed that he's never said in his entire life. He will stop what he's doing and he'll tell my wife, Nancy, what does he say? I really do love you, Nancy. It's like, honestly, every day it's amazing. So the whole reason I'm talking to you here is because just don't give up. You know, I might get emotional. Don't give up. I wrote this down because I couldn't forget this, because this is for all the families out there. If you're out there online listening, there's this really important key to this, and it's not the device, it's not the tools, right? It's the person behind it. So last thing, all right? The right environment with the right support, a tiny spark of curiosity can change everything. So don't give up. He's 29 and he's saying things he's never said before. So
So that is absolutely beautiful. I will also say, do not let your physician give up or say that your child or loved one is not capable of things just because they can't answer a question. I think and I'm guilty of it too as a physician, you look at someone who's nonverbal and assume that they can't understand. Yeah. And I think a lot of physicians do it, and I think it's important for families to remind, when you go into those doctor's visit, this is how my child communicates, whether they ask you or not.
Any other questions? Sorry.
No, thank you. Give me a. We all just met Ari recently, and he's been phenomenal. He's doing videos and stories and social media. I mean, he's a filmmaker. He's amazing. But yeah, so thank you, Ari. Janelle, and I want to acknowledge Janelle for all these handouts and these flyers. She has been designing these for me, and I think they're phenomenal. She's amazing at it, so thank you. But I asked Janelle to do a little bit on IEPs. This is just a sort of overview and what she's experienced. And anybody that has any input on this, please join in, because it's another odyssey to navigate. So keep it up to your mouth.
So I do want to follow up a little bit with what they were saying. My daughter, as she's smiling at me, recently the school did a psych test on her and told us she has an IQ of only 39. That's really low and really inaccurate. So we are fighting still, because we know that's not our child. We know our child knows a lot more than she can communicate to the rest of the world. So we're just trying to get people to see that. So that's a part of our IEP journey. It has been about a year fight now, probably longer than that. She is going to have to repeat kindergarten again, just because she didn't have the right accommodations the first year. So I'm fighting now to get her a 1-to-1. Because with that IQ score, they want to put her in a separate setting classroom. And again, that's just not for our child. She's very social. She loves interacting with other people. So we want to keep her in the regular ed classroom for as long as possible. So in the process of fighting with the school, there are just a few things, I'm going to skip over most of it in interest of time, that I think are important, and things that I have done personally that have helped me. So again, you are the expert on your own child. We have to fight for our kids. We know what our kids know and can do more than anybody else in the entire world. So we have to fight for them. And your voice does matter. At first, I thought I was just at IEP meetings to hear what they had to say about my kid and sign the papers and agree and go on with life. But I was really wrong, and I realized that, and now I have to fight for everything. Build relationships. I am also on our district's special needs council, so I am helping empower parents getting the same knowledge that I am currently learning, so that we can advocate for our children. Document everything. This is my favorite thing. For real, document everything, every phone call, everything you possibly can. Either you follow it up with an email, like, per our conversation, this is what we talked about, or ask them to do it. The other thing is in the minutes. All IEPs have minutes. If there's something you don't agree with or something that they did agree to, make sure you have them put it in the minutes. At our last IEP meeting, I asked multiple times to have things put in the minutes. Like, all her therapists, all her teachers were all saying, yeah, she needs a 1-to-1, but then the principal said no. I wanted that in the minutes. I wanted people to know this isn't just a mom thing. This is everybody who sees her every day agrees with this. So definitely you can ask to put things in the minutes, and I think that's really important. Don't be afraid to question the decisions. Again, they wanted to push her into the separate setting classroom, and we said, no, that's not right. That's not what we want for her. Oh, ask for what your child needs. So we recently got a new EC director in our district, and she's very parent empowering, and I appreciate that so greatly. And I've talked to her about Whitney, and I've talked to her about a few other children that I'm aware of, just because I work in the school district. But IEPs have the ability to be creative, so be creative. Recently, Whitney was in the back seat of the car with our caregiver, and she was eating ice cream with a plastic spoon, bit through the plastic spoon. She knows the story. And choked. We took her to the ER, to make sure everything was okay. I went in to talk to the EC director, and she's like, I heard what happened with Whitney and the plastic spoon. They used plastic spoons at school. She's like, we're not going to have that. We're going to write in her IEP that she cannot have plastic spoons. Not something you would think that needs to be in an IEP, but yeah, I don't want my kid choking on a plastic spoon again, so please, let's do something. So again, just ask for anything. I always thought there was like a list too, like here's what we can offer you, but no, you have the ability to be creative. If you think something's gonna work for your child, just ask for it. The other thing I've learned through talking through different advocates and lawyers too and everything is the parental statement drives the entire IEP. So what I did in the middle of the year is I wrote like a 2-page parental concerns, and they had in the IEP meeting they had to go through every single concern that I had and address it and make sure that it was being solved. So that's not something I ever knew. When they were like, oh, do you have any parental concerns? I'm like, yeah, I want my kid to learn. You know, I didn't know, and they expect that out of you. They expect you to be put on the spot and say, what are your, concerns? Don't let them do that. Write it before that. These are my concerns. This is what I want to address. I even send it in before the IEP meeting so they know what to expect. And then they have to address everything. And again, it's written in the IEP, so it's a legal document, so they have to address it. I guess that's really all. I can mention a little bit about guardianship. Okay. Yes. Yes.
Can I say one thing?
Yes.
I've never been on the parent side of the IEP meeting because Kinsley's only 4, but I taught for many years before I started staying home with Kinsley, and I sat through many ARDs, and the parents that were most successful in getting the IEPs to be exactly how they wanted them was they said, this is my concern, how is it affecting my child in the classroom? And then that put me as the teacher saying, oh, well, it impacts them this way, and then the team had to do something about it.
This is my sister, and she is in the school system and works with the kids.
Yeah, I work in Maryland, and I've been teaching for 24 years. And I've been a special education teacher. I'm dual certified. I teach inclusion a lot. And I've sat in a lot of IEP meetings, and I know how the system works and doesn't work. And when you were saying to advocate for yourself, that is absolutely critical. The minutes, I think that it's a tricky one because I don't think people realize that you can ask for the minutes in the meeting. If they don't have a printer in there, the printer is just down the hall. So if you want to take the time and review those minutes and then say, but wait, I have something else on my list, then you should absolutely do that. And if you leave and want to call another meeting, whether it be virtual or in person, because you forgot something or you thought of something else, which is what we're going to do when we leave, you should absolutely advocate for yourself and your child and do that. And the part you said, it's Janelle, right? Yeah, Janelle, on the parental concerns, I didn't know that part of it, so that was enlightening. So is that something that's, I know they come in, parents come in and they sign this, they got their parental safeguards and all that stuff. Is that what you're talking about?
What do you guys use for your, what system? We use ECAT.
I don't know what it's called anymore because I don't do the IEPs. We have facilitators.
So we have a system that is called ECAT, and in ECAT, it's one of the questions they ask you every. Oh, sorry. So we use ECATs in North Carolina. That's like the program that runs the IEP. And when you're going through the meeting, it says, those are the first 2 questions. What are your parental concerns? What are you concerned with your child? And then what are your goals? Like, what do you want for your kid? And that's how we start every meeting in North Carolina. I don't know, I guess that other states could do it differently, but that's just how I know we do it.
Yeah, it's done differently in Maryland, but I think that those 3 pointers are excellent pointers for parents to go in knowing, because I know that, I mean, it's all governed by one main, unless something's unchanged, the IDEA law, right? So it's governed, we're all governed by that, and there are legal obligations, but schools will not offer anything. I'm sure you know that. We're not, as teachers, allowed to suggest this. We're not even allowed to suggest, go get your child tested, ask for testing, even when we see general learning disabilities. So, at least not in Maryland. So, it really is self-advocacy, and I appreciate everything that you said. I just wanted to reinforce that about the minutes especially, and if you have something else, because you're going to go away, have thoughts, and you should absolutely call back and go back or do a virtual.
I feel like the minutes too, I could be wrong because I do not know the law completely, but I do feel like receiving the minutes is part of the law.
So it is part of it, but typically they're handed to you and then your meeting's over. So, but there's nothing wrong, just like when you're buying a house or a car and saying, well, I'd like to sit and read through these, because you're entitled to the time that you need. And they will schedule an IEP meeting for half an hour. If you think it's going to be short, an hour, 2 hours. I've seen them go on many hours if necessary. So,
yes, ours are usually like 2.5 hours.
Thank you.
Thank you.
And can I just say one more thing? It's not necessarily IEP meeting related, but just in general, I think that a lot of physicians and people that don't know your child do not know what your child's capable of. So doing short videos, this is my child's baseline mobility. This is my child's baseline, or loved one, sorry, communication style. Like, those videos are very helpful in an emergency setting. When you go to the doctor's office, and let's be honest, how many doctors get your children up on the exam table versus putting a stethoscope in a wheelchair? You know what I mean? Like, I think having little videos, like I was thinking about you saying, my child's IQ is not 39. Well, so then, like, showing short videos, look, this is how she communicates. This is her reading, or what, you know, I'm being exaggerating, but like, this is, those things are incredibly helpful in so many different settings, whether it's in a medical setting or a babysitter or a caregiver or someone, because you know, people don't believe you. When you say that's not her IQ, they say, okay, you know.
Yeah, we got enough.
So I know that Michele needs to wrap everything up, but I just also want to add that within the school system, as you know, we have specialists. So we have psychologists, and we have IEP facilitators, and we have special ed teachers, and all. And the psychologists, for example, when they're doing the IQ testing, they don't know anything about your child. So Janelle and I, did talk about how that all went down, but nothing wrong with a second opinion also.
And I agree with what you said, recording videos. I mean, sometimes I have a hard time explaining things and words come out backwards. I'm talking to even physicians, and I'm like, I don't even have to tell you, here's a video. And they're like, oh, okay, I see what you're saying, you know, and just explaining to them, the communication barrier sometimes. I know what's in my brain, but I can't get them to receive it.
I saw your son in the chair, assumed he couldn't walk. Right?
Yeah.
You know, like, so unfortunately, we jump to these conclusions and stuff, and I'm now seeing all the things that he can do. But if he's sitting in my chair in my office watching an iPad, you know, like, I'm going to jump to conclusions, unfortunately, embarrassingly.
20 minutes, yeah, because it's a very short visit. All right, guys, we are over the time, but I'm hoping that this session this morning was informative. We covered an awful lot, and we got a lot of great information out of it. So thank you to all the speakers that are still here, and all the families that joined, and all of those you virtually. We'll have this available for recording.